Hey everyone, welcome to a very special episode of Growing with Fishes podcast. Sorry about the technical difficulties we had there in the beginning. That was partially caused by me. We had some technical issues in the beginning, and what happened was I thought Colin here got stuck from Mammoth Microbes, got his thing got stuck, so I kicked him out, and then he couldn't rejoin, so we had to make a new URL real quick. So I do apologize about that. We have a really special episode today. We have Colin from Mammoth Microbes, and we have Jared from Quest Hydro. He'll be with us in a second. Everyone's just switching over from the other link. I just realized he might not have the new link. I gotta send that to him. Sorry about that. Anyways, thanks a lot, Colin, for joining us. Would you like to tell us a little bit about your company and what you guys do? We've talked about your product quite a bit.
Cool.
And a lot of people here use it in their aquaponic systems. And I know you got a couple of new products coming up as well, but why don't you tell us a little bit about what you guys are about and what you guys do?
For sure, absolutely. I'll take your lead. Just the background for me. Hey, everyone. My name is Colin Bell. I'm the co-founder and chief growth officer for Growsentia. And our company, Growsentia, are the makers of Mammoth P. I think it's more interesting to kind of talk about the background. My background, I was a research scientist from Colorado State University. I have a background in plant microbe interactions. And I spent many years in academia just thinking about microbes and how they facilitate plant growth and interact with plants. I have a lot of publications, as well as my two co-founders who are still at the university, at Colorado State University here in Fort Collins, Colorado. And at one point, we started thinking about ways where we could actually bring value to farmers. I had an aha moment one night late when I was working on a paper, and I was thinking, hey, wait a minute. And I was, intending to, bring this knowledge to the field, so to speak, and thinking about it in some depth. And again, it was probably pretty late at night. And I thought, man, the farmers who I'm trying to help with my research probably aren't even reading these papers. And I said that kind of curiously, then I kind of realized for sure that was probably the case. And so we started thinking about ways that we could actually make products in the lab, even while it's still at the university, that we could help farmers with to improve their crops. And I thought about this a lot as a microbial ecologist, thinking about more natural ways to grow crops and kind of bringing nature and natural processes back to agriculture. And so we started looking and thinking about different problems with current agriculture management. And we found that, nutrient delivery, and we kind of knew this already for sure, was a huge issue for a couple different reasons. One, plants weren't getting the nutrients that were necessarily applied to the soil at the rate that they should. And then this buildup of nutrients in the soil year after year after year can accumulate for a decade or two. And then it causes huge, algal bloom, water pollution, soil pollution. And we knew this for a long time also. These heavy salts that continue to get added in the soil year after year, among other disturbances, really reduce microbial diversity. And the microbial diversity is what actually facilitates natural plant growth. And so we thought, wow, let's come up with microbial solutions to potentially one day reduce fertilizer inputs, but mostly to mine these nutrients that are absorbed or stuck or unavailable to plants to maximize bioavailability of plant nutrients, maximizing plant nutrient uptake, and to help farmers deliver nutrients to plants naturally. So our first product, I think I have an example here. Yeah. Mammoth P. Here it is. If anyone hasn't seen Mammoth P right here, is a liquid organic microbial soil additive, or additive in general, that targets phosphorus and micronutrients, making those nutrients bioavailable to maximize plant nutrient uptake, and it actually significantly increases plant yield across a lot of crops. We tested it across a lot of crops, and we still do. Here in Colorado, I was very interested in testing on cannabis. I had a lot of friends outside of the university that would help me with that. And we saw such good yield increases on this crop in particular that we thought we would, it compelled me to leave the university, which I did in March 2015, to start a company to focus on bringing these natural solutions to cannabis growers. And so here we are now, about a little over 2 years later, and we're actually selling Mammoth P in over 40 states, and in DC, in Puerto Rico, and Colombia. And we've talked about a lot of places we're expanding into. We're expanding into the UK and into Spain this quarter. And it's been a really interesting success story for us, because mostly it allowed me to understand how to take an idea and to actually use the market, so to speak, as a tool to get solutions in the hands of farmers. And I'm really happy about and kind of grateful I've been able to take this journey and experience what we've experienced so far.
That's awesome. Um, can you tell us a little bit more about how important, in terms of the future of agriculture, it is for people to, focus more on microbes as compared to mineral salts? I know there's a ton of different issues involving runoff and, traditional, heavy mineral salt farming. I know that, what you guys have helps mitigate that a lot by utilizing what's there, and how that, generally it's kind of the way of the future. Do you want to talk more on that a little bit?
Yeah. So I've thought about that a lot, especially since we started thinking about being more applied scientists. And in general, we think, I think I'll go all the way back to the Green Revolution, and that this first Green Revolution was focused heavily, surprisingly enough. It was focused on improving agriculture yields, to meet the demands for growing population so we could feed the world. That's a good thing. But what it was focused on was three key areas: plant genetics, improving plant genetics to maximize yields through genetic breeding, etc. Through more precision agriculture, we're thinking about, being more water efficient, etc. And I think of these as good things more than not. And a third part of that was, which is questionable on how good it is. It's effective on some degree, is petrochemical fertilizer production to deliver the macronutrients and micronutrients to a lesser degree for plants. And at the time, it was a great solution because a solution was needed. And I know the minds of the time felt like this was a great scalable solution, because you can produce fertilizers to use on agriculture across the world. So it's scalable. And fertilizers, and these petrochemical fertilizers, are pretty consistent, which is important for production, and they have long shelf lives and etc. But what we find now, and actually, it's funny, I was looking back the other day and I found an article in the New York Times back in 1977 when one of the journals was like, man, these petrochemical fertilizers, we're starting to have some huge issues with these. And so for a long time, we've known that there's been some pretty serious issues with adding these chemicals into our soil, and it continues to become increasingly apparent. It does a couple different things. Just the production of petrochemical fertilizers is a pretty caustic procedure as far as environment and pollution, and that's pretty well known. And what we know also, and what I kind of alluded to, and when I was first talking, is nutrient use efficiency is elusive, especially in soils. Meaning, these farmers, and because of their management, how they've been taught, add excessive amounts of nutrients just so they can deliver as much as they possibly can for the plants. But what ends up happening is those nutrients aren't all delivered to the plant. It's just impossible to get 100% nutrient use efficiency, no matter what. Nutrient use efficiency being defined as, if a farmer has a certain unit of fertilizer that he buys and adds into the soil, 100% efficiency means the plant takes up every bit of that. But because of the chemical interactions in the soil and whatnot, a certain amount of that remains in the soil or leaches down beyond the reach of the plants. And what we know also is those, as I mentioned, those build up year after year after year, and it causes a lot of issues. But I think the ones that I'm focused on, I'm most qualified to kind of comment on, are the issues with reducing diversity and microbial life and soil life in general. as you change the chemical characteristic of the soil by adding all these ions and petrochemicals, microbes respond dramatically. And I've written a lot of agriculture-related papers. I had a postdoc at the USDA at one point after I got my PhD, and we focused on this stuff, and we looked into it. We're like, wow. You look at natural soils, and natural and native grasslands are one background comparison, because agriculture soils used to be these grasslands that are relatively stable, have high diversity, have high carbon, and are relatively functional, meaning there's nice, healthy ecosystem cycling and productivity, although it's a grassland. Once you start looking at different aged agriculture systems, meaning the amount of time they've been farmed, you see less and less diversity, which is kind of fascinating. And so it actually really prohibits nature to help these plants grow better. And there's a lot of disturbance factors. There's not just the addition of chemical fertilizers. There's tilling, which is an issue. There's monocropping, which is an issue, and all these things kind of contribute. There's not one silver bullet. There's a lot of cumulative things that add to decreasing the ability of farmers to maximize their yields. And what is happening is a lot of these management practices, including the addition of fertilizer, is decreasing the potential of these soils to produce healthy crops. And so,
do you mind if I ask you a question real quick?
Yeah, sure. Thank you.
Yeah, so
jumping in there.
I hear everything you're saying, and I think that's great. Definitely like the monocropping and no tilling and all that stuff. So is there a specific reason that you selected phosphorus, for instance, for your first product to target that particular nutrient in general? Was it work that you'd already done, or did you feel like it was the best opportunity? Because it is, I guess not as prevalent as some of the other nutrients that we would add in most of our inputs. So was there any thinking going into, okay, well, we know what we want to do, and we know microbes, and we know we want to make this more available, but was there something about phosphorus in particular that made you want to target that?
Sure, yeah, there actually was. And you got to know, when we thought about this problem, phosphorus, and its absorption desorption capacity is what we came down to understand. And we were talking to a lot of farmers and through the Midwest in particular. And what we realized is that phosphorus was being added at excessive rates, and up to 70% of the phosphorus farmers were adding year after year weren't being taken up by the plants, but they were being sorbed. Phosphorus is chemically very sticky, and so it will sorb to a lot of the clay aluminum ion complexes, and it'll sorb to a lot of calcium complexes in more calcareous soils or more alkaline soils. But bridging the gap between being a basic scientist, a basic research scientist, and thinking about more applied approaches, phosphorus is a little easier for us to think about. We saw this problem. Phosphorus is a little less complex than nitrogen, for example, and we needed more time to think about that because nitrogen transforms in several different ways, and we actually have a lot of expertise in our team, and I'm not the foremost expert, but I understand it well. But then, our postdoc that was helping me at CSU, Peter Boss, and then Matt Wallenstein, one of my co-founders, all did dissertation work on nitrogen and understood it very well, understood it enough to know that we needed to think about it more, because there's a couple different things that are going on with nitrogen as it transforms between nitrate and ammonium and ammonia and nitrite, etc. And so there's a lot of other things. There's a lot of different traits to consider targeting, and we didn't know which one to exactly attack. Phosphorus is relatively simple. It's phosphate, and we knew how it sorbed, and we knew how we could desorb it, and we knew how microbes desorbed it from these particles, or we knew how microbes actually mineralize it by creating enzymes. And so we had a microbial trait, so to speak, that we could really conceptualize well and focus on. And when we think about these products like Mammoth P, what we want to do is maximize the microbial ability at the community level to facilitate phosphorus bioavailability through those two mechanisms. Did that answer your question?
Yeah, I think so. I mean, I guess I kind of already assumed part of it. I just wanted you to talk about it a little bit more for the people that are interested in, and especially for our aquaponics crowd, obviously, since we do a lot of the nitrifying bacteria, and that loop that you talk about sort of gets incorporated into our systems a lot. Anyways, I think that it's a similar application to phosphorus, which I think is really cool. And I've always been intrigued by your product ever since I got my first sample a while ago.
Cool.
And so just wanted you to share a little bit of thought behind that. And so, yeah, I definitely think that that selecting those different things, like in phosphorus in particular, where it's supplemented so often in ways that it's not terribly effective, that it's is, in my opinion, a great product choice to start off with as well. So kudos on that for sure.
Thank you so much. More than anything else, and the way it complements the cannabis industry and applications there, it was true serendipity. I mean, we were actually thinking about broad agriculture and thinking about soils, and so we had a lot of testing to do after we actually had a product, and we tested on a lot of crops in the greenhouse and in the field, and with different soil types and different medias. And then cannabis was a whole new leap of faith. We're like, okay, we'll test this. And my early hypotheses were, because I was thinking more about soil and broader agriculture, that maybe it would work, maybe it wouldn't, but I didn't have high confidence. And when we started seeing really good results, and we could repeat them multiple times, we're like, oh, cool, it is applicable to some soilless medias. It is applicable to some hydroponic systems, where I had my reservations early on, but I think scientists also are trained to be pretty skeptical. And so I was carrying that along also.
Sure, for sure. So yeah, I think that like, especially as far as like aquatic systems, it's, like might be, again, different than what you're trained to think as a scientist, when you're always looking for optimal situations for it to survive in, or different things like that. But, I've been adding it for, quite a while to my aquaponic systems here, both with and without a soil layer, which I definitely think that, Steve's a big proponent of the dual root zone, in having that separate soil layer for it to be able to establish a different microbial profile, which I definitely feel like I've seen an advantage to as well. Do you feel like there is anything that we can do in our aquaponic systems that would help these particular microbes survive? Is there an environment, like anything from like water temperature or inputs that we can add that would be a good food source?
Yeah, so that's a great question. I'll kind of go back to, initially when we were testing in hydroponic systems, I just want to keep it to an aqueous application for a second, so I can tell you my experience. I hypothesized that these microbes would not be a good solution, and mostly because I hadn't tested it in any kind of hydroponic environment yet, and these microbes were collected from soils across the country, because we wanted just to start with the most biodiverse soils possible to select these functions that I was talking about, to liberate and make phosphorus bioavailable. And so what we realize is these microbes are really robust in aqueous environments. If the shelf life on Mammoth P, and this is in a liquid suspension, is at 2 years right now. And it's actually really amazing. And writing a scientific paper just on that, it sounds kind of lame, but it was one of the feats I never thought we would ever reach, is the shelf life of over a year, or even over 6 months for any kind of soil microbes in a suspension. And we achieved that kind of by accident. We have a platform and an IP, and the patent's public, and you can look it up pretty easily, and I can share that information with you as well, where we have a functional trait selection platform. When we think about phosphorus cycling as a trait, and we want to train microbes at the community level to improve their ability to cycle phosphorus. And so I've kind of talked about that, and generation after generation, we would select a winning community, and we'd repropagate that 1,000 times until we continue to see growth. And as we see these microbes' functional aptitude increase, we would select the winner, and and continue to propagate that over time. We see this functional asymptote, which you would expect in any kind of biological system. And at that point, the function was about 30 times greater than anything we saw in nature. And it was like a success. We're like, cool. In retrospect, after we started testing it, I had to kind of reflect on why we're getting such great results in hydroponic systems, and why are we seeing great results in aquaponic systems, etc. And when I was reflecting, I kind of had an aha moment also. It's like, wow, duh, through that selection of thousands of generations, the selection events, to get to the microbes that we have now in Mammoth P, there's also an environmental selective pressure where we used an aqueous environment of high ion concentrations, mostly to make it easier to transfer, but then to have a substrate for them to grow on, obviously. And we started off with tens of thousands of microbes in this platform from the soils that we collected all over the country. And, over tens of thousands died out, and they died out for two reasons. One, because we intentionally selected function, in one replicate of 1,000 of function, generation after generation. So inherently, we're going to leave out some of the microbes. But I also hypothesized that many of those microbes died out because they couldn't persist in a high osmotic pressure, high ion concentration environment, which is intuitive because these microbes from soils haven't evolved to do that. And so what we ended up doing is environmentally selecting the microbes that not only could function at a very high rate, but that could persist in the environmental selective pressures that they would be exposed to in management practices, such as hydroponic systems and such as these aquaponic systems. So sorry to rant on for so long, but really, I think that the microbes, if you add them in an aquaponics environment, will persist very well, just because inherently they live very well in an aqueous environment. And they're flagellated also, it turns out, which means they're mobile, and they'll actually swim around in that environment. And if there's a low oxygen gradient somewhere, which inherently there's going to be in a water column, they'll make it to a higher DO, dissolved oxygen area, to absorb that oxygen. So there's a couple things that are in their favor physiologically, which allow them to persist.
That's awesome. Thanks for all that information.
Sure.
I had a chance to talk to one of your reps at one of the Indo expos, and he was telling me that your product also increases molybdenum uptake. Do you want to touch a little bit on that?
So yeah, I mean, in brief, it's not like I've looked at it a ton, but early on we looked at nutrient uptake for a lot of the macro and micronutrients because it was interesting, and we did it across a lot of different plants. And what we're looking at in those particular studies for sure is just the mode of action that solubilizes or desorbs phosphate. If you have a phosphate molecule and it's stuck, it can be desorbed through ion exchange, which is very, very common. But that same function or mode of action can work on a lot of different ions. And so we saw higher iron uptake. We saw a lot of micronutrient uptake that was, and how we measured that was obviously we looked at leaf tissue nutrient analysis of the macros and micros. And we saw a higher calcium uptake. We saw higher iron uptake, et cetera. So although we were targeting phosphorus, what you can't do is make biology just do one thing. I mean, you can't shut everything down. And so, these microbes actually produce enzyme activities that facilitate nitrogen cycling. They facilitate some carbon cycling. They facilitate phosphorus cycling very well. And then through the absorption-desorption capacity of this community, they can actually liberate a lot of these metals and micronutrients as well.
We had a question from chat. It says, when is the best time in the plant's life to use Mammoth P?
So that's a great question. What I recommend is, we always recommended, at least from the very get-go, bloom for sure. Start inoculating at the last couple of weeks of veg and carrying it all the way through bloom. And I did early studies where I actually looked at the effectiveness during the clone stage, veg stage, and through bloom. And what we saw even early on, and growers are still seeing this, is anecdotally, people are reporting all over the place. They see a lot more clone vigor in early stage clone, but quantifiably what we're seeing is more root development and faster roots. And so that's a really interesting value. If you're getting clippings or seedlings, and you can get more root generation early on, because it just carries through to the health potential of the plant throughout the whole life cycle. And intuitively, that makes sense biologically, because these plants actually hunger for additional phosphorus during that root development cycle as well, early on in plant growth. And so a lot of growers started at clone and just carried all the way through. I recommend at least use it in the last week or two of veg and push it all the way through bloom. But I think your growers are seeing really, really good results who use it all the way through as well.
Awesome. So we have people often debate whether it's better to use Mammoth P directly or put it into your compost tea when you're brewing it up in order to dose with the other microbes. Which do you have any kind of opinion on that, or which would you say is the better way to go?
Yep. So I think that I'll comment on compost teas. I love the practice of using compost teas. I think it's a good idea for sure. And when people ask me that, and they ask me that quite a bit, what I recommend is, if you're going to use your compost tea, to go ahead and brew your tea so you can maximize the diversity that you want to brew, and then add Mammoth P at the recommended application per volume at the very end of the brew, because the microbes in Mammoth P are active microbes. They will grow. And I at a more concentrated rate, most likely, than you need them to if you start them at the beginning of the tea. But if you'll add them at the end of the tea, you can actually keep that application rate dialed in and just simply add the function of Mammoth P to your compost tea.
Oh, good. I haven't been telling people wrong then.
So I understand you have some new products you guys are working on. Do you want to talk a little bit about that?
Yeah, we're always innovating for sure, and we don't have anything ready to release yet, but I think it's very interesting, just as an overview, to have prescribed microbial inoculants to meet plant growth nutrient demands. And so I think it's really interesting to think about deploying maybe a Mammoth N. We kind of alluded to that earlier. YP, potassium's interesting for me, for sure. Maybe some micronutrients. Right now we're working on a Mammoth N product, and we're working on a Mammoth K product, and it's still in the R&D pipeline. And just so we're clear, I'll explain the R&D pipeline just briefly. We have these different segments. We have the development phase, which can take forever if you never get through it, but we're in the development phase right now for sure. And then we have the lab validation to make sure that we can actually quantify the modes of action, at least at the lab setting, that we think will allow these microbes to deliver the value and the benefits to plants. And so that's the second part of the R&D pipeline. The third pipeline is the plant testing pipeline. And we have greenhouses that we control, and we're growing plants and a lot of different kinds of plants. And we have some outdoor grows as well. And we make sure that we get those technologies on plants and they deliver the values that we hypothesize they will. If we see those positive results on those products, and then we'll get it out for the beta testing with some of our partners. And if we still validate those results, we'll take it to the next level and start getting it in more people's hands.
Very cool. Any plans for a Mammoth Silica?
We've thought about that a lot for sure. There's only so much you can do at any one time, and so do you think there's a lot of interest in that? Like a microbial, microbial silica product?
It makes an insane difference, especially in aquaponics with reducing molds. Like night and day difference.
Well, one thing I think, I haven't met a single grower that doesn't use a silica product. I mean, everyone uses a silica product. Now, I was still trying to kind of conceive how I could. We're definitely a microbial company, and we're never going to be a fertilizer company, and we're never necessarily going to be just a simple a nutrient or additive company. We're going to be a microbial additive company or a biostimulant additive company. And so I'm trying to figure out how to marry microbes and microbial processes to maximize the availability of whatever we're trying to deliver. Because at the end of the day, it is nutrients, but there's a lot of benefit in incorporating the microbial process with that. And so it came up a couple times, I'll say, Steve, but it's not on the top 2 right now.
Steve's he's big on silica. It brings it up often, let's say.
Got
So
it.
Steve has a lot of interest in it. He's trying to get more. But
Steve, it just got pushed up on the list just because of that, okay?
And I actually started adding a horsetail fern tea to my plants a couple of years ago and really saw a big difference in it as well. And there's a company, I think it's Tea Lab out of Eureka, California, that makes silica solubizing microbe inoculant.
Oh, cool.
Which
sounds cool. Is it a good product?
You know, I tested it last year and not very effectively because I just basically added in with a bunch of other stuff. So it's all, I don't know how much difference it made independently, but it definitely, I added it to a couple of batches of horsetail fern tea and saw it definitely increase microbial life and like a thicker mat on the top and different things like that. But nothing where I did like a grow off or anything like that. But I do think that they make a lot of good products, and we talked a little bit about teas earlier, so that's definitely one of them. And they're really big in the Probiotic Farmers Alliance group. I don't know if you are familiar with that group or not.
Yeah, I'm a little familiar with it. I'm not actually I'm not in that group, but I know of them for sure. Where are they out of?
Facebook group.
Tea Lab, they're out of Arcata, California, I believe.
Cool.
I think so. I think that's I can't remember his name. Luke, I think is his name. He posts in the Probiotic Farmers Alliance a lot,
Cool.
which is
I'll hit him up. Thank you.
on Facebook. And one of the questions that come, because you know, Mammoth gets discussed in a lot of different cannabis forums, obviously. And one of the weirdest questions that I come across a lot, or more recently, is people keep coming up. They bring up these articles that were posted a while ago that talk about some of your microbe strains as being possible human pathogens. And when you really get into it and read it, the first thing that all of them say is that it's well documented as not being a human pathogen. But how do you, how should I or anybody else respond to somebody who I feel like interprets something that way?
I think that's tricky. It's a slippery slope for sure, because, you know, I read a couple of those papers too. And, you know, the last thing I want to do is, first of all, deliver something that's going to be harmful. And second of all, to have people scared about a product. And I think that the misinformation is really unfortunate. And I think it's just a disconnect in some understanding. If you'll see, and what I'm assuming is people read a title and it has the name of the bacteria in there that says something bad, and you're like, oh God, this does this. But and like you said, I really appreciate you saying that you read the paper, because when I read the paper too, I was just kind of ultimately frustrated. I was like, this doesn't say that at all. It's just not true.
Right.
And, you know,
no, it doesn't. Yeah.
We have a, you know, really, not only are we, you know, pretty well-known scientists globally, but we have an advisory board, and we're really rigorous on the microbes we selected. And, you know, honestly, you have to be, because you have to go through Department of Agriculture for every state, and they have lists of microbes that you can't register or use. And so we go through these very, very rigorous processes. We go through the, you know, OMRI certification. There's a lot of checks and balances for microbial products for these particular reasons. And we knew the answers to those, because we looked at, you know, the biosafety level of all the microbes, and you just can't market microbes that are dangerous like that. But people, some people more than others, get concerned, and in the social media in general can get a little out of whack. And I'm at that age where I'm a little older. I really encourage anyone who has any questions and they want to talk to a scientist about this, Colin@growcentury.com. I'll give you my direct phone number. Let's talk about it, because I don't want anyone to be concerned about our product. I don't want people to feel scared. And I'm afraid that some of these flare-ups is just kind of fanning fear. And I don't appreciate that at all, because I don't think it's true. So please reach out to me. I'll be happy to talk to you about that. And to answer your question, it's really hard when people are worked up to sway people's opinions. I try to support a clear, consistent message and make people feel at ease that, these are kind of the facts. This is really well known about these microbes. And I would love to walk people through these papers and show them exactly what the study's doing and what it's saying and what it's not saying, if it would help them. We're here to educate. We're here to support,
Right, and a lot of them weren't even like the ones that we're referring to. I don't even believe any of them were studies. They were suggestions for studies to be done based on a theory about why somebody died that they couldn't explain otherwise. And the presence of this bacteria was essentially something that somebody suggested could be a reason for it, because they couldn't find anything else. Was the main article that I feel like it's a very deceptive title, because like you say, it includes the name of it. And I don't know if that's the same one that you read through, but it's like the third one or so that pops up if you.
Yeah, and they're all older. Yeah, I know.
Yeah, and they're all older, and none of them were really followed up on. And so I wanted to give you the floor to sort of discuss that and at least say, hey, read them for yourselves, because the title is misleading. And if you see somebody repeating it, and I questioned myself whether or not it should even be brought up on the show, because in a way you do give it the floor and allow other people to know about it. But I feel like it's been popping up so consistently now, like whenever somebody mentions it, it gets mentioned again. So that's why I wanted to bring it up and give you a chance to respond.
Thanks, I appreciate that. And I think you said it for yourself, let's read these papers, or let me read them with you. Some of them are kind of tricky to read, or just misinformation can be spread so easily. And I'm guilty of spreading or repeating something that I heard. And I think that's where some of this perpetuates too. Someone says something, you just kind of repeat it without really even understanding. But at the end of the day, we're very confident, and we put a lot of thought and into what we're using for agriculture, soil amendments for sure. And we stand behind it 100%.
Again, right, obviously, like you mentioned, to get your product to shelves, it has to go through rigorous testing. And on your website, you guys have a lot of safety information that's already posted. And so, I encourage people to give that at least as much merit as you give a 20-year-old article.
Thanks.
Because it deserves that much, right? So I just wanted to be able to discuss that, and when it comes up, then I'll be able to link people back to your answer here and allow people to see it and your offer to have it discussed, and at least you'll have a say in the argument.
For sure, and we're going to continue to post more information, at least on our website. And I've made a point also to have a nice maybe autobiography for every one of our microbes, and we're compiling that now. Very nicely peer-reviewed autobiography of any species I'm ever going to use, and so people can at least look at that. And I want to make sure it's palatable and easy to read. And at the end of the day, we have a great product that works really well, and it's intended for its intended use. And it's unfortunate that some of that happens, but I don't really lose sleep over it, because I don't feel like there's any validity to it at all, because I just know there's not. But it does hurt my feelings sometimes, just so you know out there. And sometimes when this is my baby for sure. Left the university, and for sure early on, when I heard that the one time, my stomach kind of jumped up to my throat, and I was like, oh, I don't. But we feel very confident where we are, and in Mammoth P. I'll just leave it at that.
It's no different than the stupid tilapia or poisonous things, because they're all in the Chinese ponds. It's like, no, Chinese tilapia raised in toxic ponds are, but it's not stuff out of an aquaponic system that's been fed good food. Like, it's ridiculous.
Right.
So We had a couple of good questions coming from chat here. What is the relationship with Mammoth P and other enzyme products like Canazyme or Biocozyme?
Got it. So, I used to use Canazyme when I first started growing. When I was taught to grow, that's what I was told to use. So I used it and I did side by sides. Canazyme, if you'll read the label, they focus on carbon degrading enzymes. And I think their stance is they want to get rid of all those little carbohydrates to minimize pathogen infection. I think I read that on the product. If I'm just characterizing that at all, I apologize, but that's my understanding of the value proposition of that product. The microbes in Mammoth P and Canazyme, let's just talk about the products are as an enzyme. Enzymes aren't living microbes. They're not even cells. They're proteins. They're functional proteins that are kind of conformed in a way that actually perform a function. Because microbes don't necessarily have mouths, they have membranes, and they absorb nutrients and food through their membrane, through their cell wall. They have to create things called enzymes that they squirt into the environment outside of their cell to break down carbon chains and food and nutrients into particles that are small enough that they can actually take into their skin, so it's size limiting. So enzymes, we'll think about them as exoenzymes, so squirted into the environment everywhere, in water, in waterways and lakes and soils, etc., to do the work of breaking down and degrading carbon and mineralizing nitrogen and phosphorus and making it in small enough particles so plants and microbes can take them up. Canazyme focuses on carbon degrading enzymes, and it's typically in a buffer because enzymes can misform pretty quickly by shifts in pH, etc. And just know structure equals function with the enzymes. So if enzymes maintain, I always do these little, oh no, this isn't meaningful. I always do this with my hands. They maintain this structure. They can perform the function, but they can get misshaped pretty quickly through mostly chemical pH shifts, and then they stop working. They turn into high nitrogen sources, because proteins are, that can get degraded by other enzymes and turn into a food source, and sometimes they reform and continue to function again. Microbes, and I'll talk about bacteria in particular, but sometimes I do this also, they're like little bioreactors, like little enzyme factories. And so these are the cells that actually continue to produce enzymes. So our product has a lot of enzymes and a lot of enzyme activities, but it's as a result of the active microbes that are in the product. And on our website also, you'll see, and there's a really bad video that I made a long time ago. It was one of the video publications of enzyme activities and how we assess that. You're welcome to watch that, but it's not very good, and I didn't do a great job, but it's pretty informative on how we used to assess that. Saying that, I'm trying to qualify myself as someone that knows kind of a lot about enzymes and enzyme assays in the environment. And Mammoth P, it turns out, has really high carbon degrading enzymes, pretty high nitrogen degrading enzymes, and very high phosphorus degrading enzymes. So it's a pretty diverse product functionally as far as the enzymes that these microbes produce. Does that make sense?
Yeah, absolutely. That was a great explanation. Thank you.
Cool. Sure.
We have another question. If you feed 4 times a week, do I have to use Mammoth P every feed, or just once a week, or alternating feeds, or what do you recommend?
Yeah, so that's a great question. What I recommend, generally speaking, is, every feed up to twice a week. With the commercial growers, especially in Colorado, that had the reservoirs, huge reservoirs earlier on that were adopting Mammoth P, they would just add Mammoth P in their nutrient reservoirs and feed it in the drain to waste, etc. And that works very well. And it's a pretty low application rate, about a 0.6 mil per gallon. And so they were happy to do that, and they see great results, especially if you're more of a home grower and you're doing these batch mixes or whatever. I recommend, up to twice a week. And if you have a lot and you're trying to scale and experiment, you'll see good results once a week, but especially if you want to do the once a week, I would definitely want to start earlier, at least into veg. We get samples out all the time because we think that's the way for growers to experience Mammoth P. I mean, that's the way it works as far as I'm concerned. And we haven't advertised. That's how we've grown, by getting samples out to growers and letting them speak and letting them experience it. saying that, sometimes we'll ask the growers, hey, when do you use Mammoth P? Because we didn't really know, because there's so many people. And we had over 1,000 people respond to us. And about 55% of them start using Mammoth P early in veg, and another 30% use it in clone. And so that's kind of the breakdown based on that sample size of when people have started adopting Mammoth P.
So where do they go to get a free sample?
You're talking to the right guy. I know a guy that makes that stuff. Let me just get your information. We have a sample request form that we process, and we'll get a sample. And I'll say, for a long time we were sending samples direct to folks, and we're still happy to do that. But we started getting some pushback. Our retail channels on our brick and mortar stores are really, really important to us, because I think it's just a staple in the industry that we want to support. We started encouraging everyone to pick up a sample at their local grow store, and in that sample request form, we ask if you'll just tell us what your favorite grow store is, and let us ship the sample there. And we like doing that, but it's not like completely mandatory.
Well,
so
in my case, I live in the country and I'm legally blind, so I can't drive. It's over an hour to the only hydro store within 100 miles.
Yep, and so in those cases in particular, that's why we definitely don't have that mandatory, and we're happy to ship a sample directly to your house, and we're going to do that for sure. I'll make sure I get your information. I'll get to the top of the list since we're on the show together. How's that sound?
Yeah, that sounds great. And we'll talk about some stuff later about it.
Awesome.
I got an online presence too, so I might be able to sell it on my online presence.
Nice.
All right. Sorry, I was late, by the way. Sorry.
No worries. That's actually how I started using Mammoth P. I ended up getting some samples.
Yeah.
I went to some grower meeting that either you or one of your guys was at down in Denver somewhere. It was like some grower meetup, and picked up a couple of samples, tried them and the thing, and then eventually tested them out at the aquaponics source. I know you gave us you guys sent us a pretty big bottle to test, and I mean, we took a whole one of your bigger bottles and put it straight into the 300-gallon fish tank in like a 500-gallon system, and had no, didn't kill anything. You can pretty ludicrously, and you should never do that. I'm not telling you to do that, but I'm saying, like, you can ludicrously overdose by accident and you won't kill your fish in an aquaponic system or negatively impact the system.
That's right, for sure. And early on, for sure, I tested that also. I wasn't growing aquaponically, but I poured a whole liter on one plant, because you have to test toxicity, and it makes sense. I mean, they're just natural soil microbes and organic extracts, so it shouldn't do anything negative. You can just kind of waste product, because it's not going to linearly let your plants grow that much bigger. Your plants have a threshold of how big they're going to get anyway.
Yeah.
You don't like just pouring money out? Is that what you're saying?
Basically, yeah. Don't just pour money out.
All right. Got it. That's the layman version.
That's right. Thank you.
Well, it works real good, because in my case, I teach people. So that's one of the things that I qualified products and all. So when I talk to people like you, and since we had 800,000 hits last month, page hits on our website last month, and so before I could tell somebody, hey, Mammoth P is a great thing, talking to you is great, but I always like to try it, and so I appreciate it's great. I think it's a great idea that you worked out some way to get people samples so they could try it and prove to themselves they wanted it, needed it, and it did the job. So that's fantastic.
Yeah, nice.
It really helps you maintain your price point as well too, because it's definitely not I wouldn't say it's an inexpensive product, and by giving out samples, it helps you maintain the credibility that I think that you want to be able to maintain the price point that you have. And so, and I think most people that have gotten samples that I've talked to, and that's the same way that I came into contact with it, is I got one of the small bottles from one of my friends. So I know and then I also got the link for your sample. So then I got the other bottle and the 2 little bottles and gave away the 2 little bottles to other people as well. So I assume it's probably something like what you guys imagined happening anyway. So, and it came in a nice little package and sticker. It was all very well done. And so I do disagree. You say you don't do marketing, but I feel like that is your marketing.
That is marketing.
Yeah.
So
effective
So
for sure.
we don't like to do paid advertising, I'll say that, but we do have that. I would say that's a good marketing cost for sure. We put all the money into the samples. Yeah.
Which is, I feel like has been effective. I feel like it's, in the people that I've talked to, because, obviously there's pushback, and I would imagine the cannabis industry probably provides you a place where people are willing to approach a product at that price point and prove its worth, for instance, as opposed to somebody trying to grow strawberries on an agricultural level or something like that. It might not, seem as attractive. So when the cannabis industry with growing a product that has such a higher price point, growers have already a variety of different ways to test it, mediums that they use, indoors, outdoors, all over the world. So do you feel like that, it sort of gave you that platform to sort of do more development, if you will?
Yeah, so I think this is a pretty fun topic for me. So for sure, we thought about a model of, one of our core value. We think about value and core value and culture, but I'll tell people around here every day, if we're not bringing value to growers, we're not doing our job. So I mean, that's what I left the university thinking. We're bringing value to growers, and one way to think about that is you can focus on some higher value crops at least early on. You can inherently bring them more value, because if you can increase yield 16% in corn versus cannabis, well, you're definitely bringing more value to cannabis growers because their yield is that much more. And it allowed us more so on the production side to scale, because I didn't have a lot of money to spend a ton of money on scaling equipment. We had pretty simple microbrew equipment to brew our soil microbes, and so it's like a microbrew operation here in Fort Collins, Colorado, which is kind of appropriate. It's a brew community. But I'll say also, I think this is kind of funny for me, because we're really thoughtful on value also. Not only do we bring value through yield increases, et cetera, but I thought about also on the alternate side, creating a highly concentrated product, which would bring a lot of value. And if you look at the application rate and the cost per application rate per gallon, it's actually at the hydro application rate, it's only about 12 cents a gallon in the liter pricing. And I was doing the math on some other products, and I was like, dang, if you'll do the math, the application rate per gallon, and it's one of the lower priced products on the market, but it's that initial price and that initial investment that people kind of focus on. But honestly, the application brings a lot of value.
And I think it's deceiving because of this, the concentration level is what it comes down to. I don't think people realize how far it'll go, even at the recommended dose or even above the recommended dose, and that it just seems like a small volume for a high price, and a sample is a great way to get over that. And I think it's been effective for you for sure.
Yeah, thank you so much for saying that.
How concentrated it is has a lot to do with that. You're right. That's a good idea. Good for you to point that out too. And to
thanks.
Whether it's pricey or not, because if it's 12 cents a gallon, I don't have a problem with that. If you get that many gallons out of it,
right. Early on, so some of our early retail partners pointed it out to us. They loved it, they got it, but they're like, they heard pushback from their customers, and they're like, calling. And early on, as a co-founder of a company, I wear a ton of hats, and we have this startup. I was the outside sales guy, so I was carving it out in Colorado, going door to door, talking about Mammoth, sending samples out to the stores, etc. But they're like, Colin, you have to get stickers on these bottles to show people how much it applies. I just want to show this. So my packaging guys hate me for it, because they have to apply extra stickers. But we got these stickers to say, hey, like this liter treats up to 1,600 gallons, because it allows people to kind of do the math a little quicker instead of looking on the back of the bottle. And it was a really good thing to add on, because people can kind of do that math now and make sense of it, of the cost.
What is the recommended application rate and dollar per application rate per gallon, I guess?
So it depends on the price of the bottle. So these bottles are tiered differently. But so for the example of the liter, and the hydro application rate treats 1,600 gallons. And the hydro application rate's about 0.6 mils per gallon. And so again, that's about 12 cents a gallon. If you parse that out into a liter. And it's obviously cheaper at the gallon, and it's a little more expensive tiered because you pay a little more per volume at the 500 ml bottle and at the 250 ml bottle, but it ranges all well under 20 cents per gallon application.
Yeah, that's super cheap compared to a lot of other products out there.
I know.
Which, again, I think becomes apparent when you start using it. When you're only putting in, a small amount per, then, and I think, again, having the cannabis industry that allows you to sort of gather the math, because it's obviously an industry that's obsessed with yield. So they're obviously going to track that already. So, and then on top of that, you have people that are interested in the other aspects, the more quality aspects of it. You just have a wide spectrum of people to carry out your testing in a way, sort of like a beta testing for software, if you will.
For sure. I'll tell you what, too, that, the cannabis industry, I am so grateful for this movement in this industry. And, we did a lot of discovery on a lot of crops, and there are so many hurdles to market to get young technologies into the hands of farmers. And unfortunately, because agriculture has been around for a long time in a lot of crops, these large agro-conglomerate companies have come in and just kind of owned it and controlled the whole market. And we found out at a pretty early stage that, for us to really be effective and infiltrate a lot of these crops, it would require partnering with an agro-conglomerate company because you just can't have success trying to go around these giants. And it's not something we wanted to do. And so identifying cannabis as the industry where we can control the fate of our technology and bring value to growers, it just gave me and our team here a really awesome opportunity to be able to take an idea from academia at the university and to deploy it in the market and get solutions in the hands of farmers ourselves. And I'm just really grateful for that, because not a lot of people can say that, especially if you're going into any of these other crops. And
right.
So,
and especially if you take now the math that you've gathered from those people to, track the kinds of yields that you've increased or different things like that, you could take those now to other agricultural markets and based on the math and say, look, percentages is percentages. So, whatever your profit is, it's going to be, 16% more or whatever it is. So I do feel like that it just gives you that math to base it on in a market that already funds itself. So you didn't, like you said, you could bypass some of those large agricultural norms, if you will, because a lot of cannabis growers already avoided them.
Yeah, I think that's exactly right. And again, it allowed us to validate the technology in a way to allow us to control it and do it our way and hold on to our culture and our values without having to, again, go down some roads that other companies may have to find success.
And 1 second, excellent. There we go. What does Mammoth P do to your plants in terms of growth pattern and traits for the people that haven't used it? Pretty much everyone on here has used Mammoth P and loves it, but for people that are out there and might not understand why they should use that or how it would fit in with their normal dosing regimen, you want to touch on that?
Yeah, so I'll just talk about compatibility quickly. What we realized when we validated it, that Mammoth P is really compatible with all management. You can just simply add it in with your fertigation or your nutrient additions. And so it makes it really easy. You just add this as an ingredient and put it in with your nutrients. If you wanted to add it in as your compost tea, like we mentioned earlier, add it in at the end of your tea and just water it in. And so it's really simple. It has to be, I think two things that Mammoth P really has going for it is it's super effective and it's simple. And it's fun to use. When I was growing, and I had some gardens that I was managing and growing when I left the university, before I left to make sure it worked, and I had to see it for myself. And I found myself having just a ton of fun using it, partially because I made it. And so I was like, oh, this is cool. And I was seeing great results. But, it's easy, and it works really well. I'd encourage you also, what we do is I've written very short and super like palatable papers on the importance of phosphorus for plants, the importance of nitrogen for plants, the importance of potassium for plants. They're on our website under the resources, and I can share them, and you guys can have these too. And they're about 2 pages each, and it talks about why plants need these certain nutrients, not just, you know, the timing of it, veg, bloom, finishing, etc., but why is phosphorus important? You know, and phosphorus turns out to be really, really important for plants physiologically throughout the whole life cycle. You know, there's ATP, which allows all cellular work to occur in humans and plants alike, and microbes, for example. And the P in ATP is phosphorus, right? The cell walls in plants have phospholipid fatty acids. The phospholipid part is phosphorus. Our DNA, in plants' DNA, has phosphorus heads on there. And so phosphorus is really, really important to incorporate into all sorts of cellular processes and constituents to allow plants to continue to grow and thrive, maintaining their genetics, maintaining their energy, etc. And so, you know, honestly, early on, I felt like growers are really going to see the benefits of using Mammoth P on the scales, you know, at the yield. And they do. I was pretty surprised when early on, and you know, we had some pretty cool things going where we were just meeting some growers, and we just meet them in a park, and they had some pretty big grows, and they were trying Mammoth P for us, and we get a call a week later saying, man, you have got to bring me more Mammoth P. My plants just responded immediately in a day or two. And one guy, we use this sometimes, at least we used to love this phrase, my bud went from wrist size to fist size, Colin. I mean, seriously, I need more of this stuff. And I took it with a grain of salt. I didn't really understand why we would see immediate responses at that time. And we saw more and more of it, heavily in cocoa, but in other systems also. And what that comes down to is, you know, I think that there's micro lockout events of varying degrees throughout the plant's life cycle when you're adding these nutrients, etc. And microbes in general, the microbes in Mammoth P, just help just maintain the bioavailability of nutrients and kind of minimize any micro lockout event. And especially if your plants, when they see the immediate responses, I think we're probably experiencing a little bit of a lock that was just released, which the microbes would do just naturally, and allowing the plants to thrive again and visually really quickly. So what I'll say for sure is what we think is you're going to see it on the scale in yield in your harvest for sure. And many growers are saying that their plants have never looked healthier, which is a little more anecdotal. It's hard to have a measure on that, but you have a feel for it for sure. And so, you know, more vigorous plants, healthier plants. We like to say also that it allows your plants to maximize their phenotypic potential. A healthy plant is a happy plant. You know, we breed our plants, and it's in the genetics for those chemical characteristics, terpene characteristics, and yield characteristics. And if you can maximize your plant's health, your plant's not only going to fight off any type of potential pathogen naturally, because plants are able to do that naturally, but it's going to be able to maximize these characteristics for which it was bred. And so you're going to get the highest quality and potentially the highest yield by just allowing these processes to occur by incorporating microbes into your grow.
Have you, we had another question from chat. Have you tested this with labs or other fermented products like lactobacillus ferments or anything else?
No, I mean, we work with our microbes in our lab at CSU, but to a large degree, we're trying to ask more mechanistic questions, like what else do we think that it's doing to help facilitate plant growth? Because we know that, you know, they're probably not doing just one thing. We haven't done a lot of mixing. We did some mixing with some other like delivery. Like I think it'd be interesting to do a dry related product, and we were working with a company for a little bit, and we're testing with biochar. And I was actually working with a company a long time ago, and they had some type of kind of funky silica. It was like early days for sure, before Grosentia, but still at the lab. And we hypothesized we could use those as carriers. Biochar was pretty interesting. That early stage silica product, which I didn't understand well enough, didn't work great. And so we're still thinking about, you know, are we going to make a version 2? Maybe someday, but I think we're way more focused on creating that second product, like we talked about, the Mammoth N or the Mammoth K. And so that's where what we spend most of our time doing in R&D right now.
Do you think part of that is because you achieve such a high shelf life with your liquid suspension already that, you know, most of the time that's why solid inoculants are, you know, favored, I guess, because they last longer than, you know, most liquid suspensions. Microbial, you know, 6 months is, you know, sort of a standard that I like to go by anyway.
Right. So yeah, for sure. That's what we hypothesized too, that we were going to get. And so since it was such early days and we were on the very early stages of the Mammoth P liquid product, and we were hypothesizing before we even had the data that we were going to have issues with shelf life, and we were looking for solutions for dry product and for, you know, some people just like having dry product. And so we weren't excluding anything. What I had a hard time and why I kind of abandoned it at the time is, you know, we can inoculate these substrates. I'm not talking about the biochar, but this first one, this silica-related product. But I could never recover the microbes off of them, meaning that, you know, I couldn't, even if we would dry it on there, and what I wanted to do is see how much, once we put the dry product back in suspension, what the microbial concentration would be when we grew it back out. And I never could get a suitable concentration back out of those particles. And it was frustrating me to the point where we were moving pretty quick. And at that stage, it was limited bandwidth. It was me and Lauren, who was the first lab assistant I had, and then Melanie was on it, and then Peter came on, and the four of us were working on this. And we had so much going on that we're like, man, let's just shelve dry product aspirations at the time, and focused on what was working well for us. And again, you know, when we launched Mammoth P, we launched it with 6 months on the label because shelf life data is real-time data. And we had 6 months worth of data on that at the time. And I was happy enough with that to launch a product, to start to get it in the hands of growers. But there was a little bit of a leap of faith. Again, when we had a year, 18 months, when we got to a year, I was pretty surprised. I was like, cool, I think this is going to keep going. And it is, you know, it's at 2 years right now. But the daunting thing about shelf life data is, you know, if you need 2 years worth of shelf life data, you almost need a 2-year experiment. And so it's hard to wait 2 years to think if you're going to launch this. So, you know, we were a little aggressive when it comes to that, and I would just say hopeful. And it worked out well.
Awesome.
Awesome.
Does anybody else have any other questions for Colin, guys?
I actually had a question. To make your product, is it a set of processes that culminate into a final product, or is there a particular substrate that you inoculate that the microbes will culture to?
Yeah, that's a great question. So what we had to do, unlike any kind of bioproduction facility, you have to make scalable media. You know, early on, we were making a highly selective media just to make sure we could control every input. Like I had every ion down where I could measure it in this media because we wanted to be very, very controlled. But when we start scaling it, first of all, we wanted to be an organic company. We couldn't scale chemicals, and we didn't want to anyway. And so I had a lot of this background knowledge already. We knew microbes grow on nutrients, right? And carbon-rich substrates that have adequate supplies of nitrogen, etc. And I came up with a mix of some forage, organically grown, locally grown forage, and feed that we could extract quite simply like a tea, like a hot tea. And then we could extract the carbon and the nitrogen and just to use that as a broth to scale up our microbes. So our microbes and our consortia, you know, our 4 microbes are the technology and are the IP, along with the platform, how we select the microbes. That's in the patent. And as we scale it up, we just do it, and we try to do it in ways that we can maintain organic certification. So we're bringing organic natural solutions to growers. And we can do it more affordably. for sure, because, you know, I think that's a big problem for a lot of these manufacturing companies, and that's why their prices are higher in some cases. And the microbes that grow it highly concentrated. And so we kind of ticked all the boxes with our mix, and we're still using it.
I didn't realize you guys were OMRI certified. That's awesome.
Yeah, we're totally OMRI certified. It's important for us. You know, we knew we were organic no matter what, by definition, natural organic. But to get the certification was, you know, an extra tick box, and that's not easy to do. I mean, that organization is pretty rigorous.
I have to say congratulations too. Yeah, that is. It's a 4-year process here. We want to get certified organic. We have to produce and be inspected for 4 years in order to get the OMRI certification. I don't know about what you had to go through.
No, that wasn't our experience, but it took about 18 months.
Oh, well, I guess Colorado's a little more progressive than Carolina.
Maybe so.
Very fair assumption.
Oh.
Hey, wait a minute. I think that is true. Actually, I think Colorado is a little more progressive than Carolina.
Yeah, never would have guessed that in a million years.
So the other thing I want to mention about the, you know, I guess the difference in your product versus like something you can culture yourself is you talked about how you selected strains as you go from sort of step to step, and can you talk about how long that process takes? Because I think a lot of people go, oh, well, you know, I hear this a lot in the PFA because they're big on making their own stuff anyway, but they, you know, they sort of say, oh, well, I can just take some, you know, alfalfa extract and, you know, culture my own. And, you know, I guess my response is normally like, you know, okay, go for it.
Show the results.
But can you talk about maybe the amount of time you guys put into sort of those, you talked about, you know, selecting generations of sort of advanced microbe, I don't know what the technical term is, but you touched on it earlier.
Yeah. So the process is, what we're able to do, because we're at the university, is we had a lot of equipment at our disposal for sure. And so we actually have transferring pipetting robots, etc. If you guys are coming out sometime, I'll show you the lab. It's really cool. I think we have some videos where, you know, it's really hard to hand pipette thousands of wells for every single deal, but we could just program the robot to do it for us. Super cool. And we had some grant money to help us fund this stuff. This was, you know, focused on agriculture innovations. I actually wrote a grant to help bootstrap and flip our lab so we could make this happen for sure. It was thousands and thousands of generations, which can take a long time. Microbes can grow anywhere between a day and a week. But to do that thousands and thousands of times, one of the kind of benefits that we had is we could do that thousands of times for every generation, and so that helped in the high throughput manner for sure. We ended up getting a product after thousands and thousands of generations, but in time wise, and I'll say we kind of got lucky, because people don't usually do this. We got it done in about 5 months, where we had our first consortia, which turned out being Mammoth P. But saying that, again, this was thousands and thousands of generations to get to that. And so, you know, you can do the math depending on your resources. If you have one batch, that's, you know, a lifetime of work.
Gotcha. That's a perfect answer. Thank you.
Sure.
Yeah, that was great. Anybody else have any other questions? B or Roger or Marty? I had one other question now that I think of it. What books would you recommend for people that are out there, or any other information or resources would you recommend for people out there looking to learn more about soil microbes or aquatic microbes, or, you know, just this whole kind of thing in general?
Yeah. So, you know, I have a lot of books actually behind me, but they're more academic and they're not fun to read. And I would stay with some books that are really, I'll just say, digestible and understandable. And Jeff, I'm sorry, Jeff, I forget your last name, but he wrote a book, Teaming with Microbes, that people are really starting to enjoy. And I went to the MJ Biz conference in DC not too long ago, a month ago or weeks ago. Time flies pretty quick these days. And he talked about his book also, and he gave a talk. And I think that's a great book to start with. You can learn about what bacteria do in nature, what fungi do in nature, what soil food webs are, and these other components of that. There's another guy that just came to Fort
So he has
Collins.
three books, right? He has Teaming with Microbes,
That's right.
Nutrients, and Teaming with Fungi. And I think
Teamwork.
Teaming with Nutrients is the one that just came out. And yeah, if you're looking for books, those three come highly recommended.
Yeah, I think so.
What's his name actually there? It looks like you got him up on search, Marty.
No, actually, I just know.
Oh, I'm sorry. Well, hell, that's pretty nice. You knew all about it already.
And so I'll say that Jeff, I'm looking it up right now, just so Lowenfeis, I think that's his name. Lowenfeis, and he doesn't really know me personally. I just watch this talk, and I support people who are supporting the idea of organic growing and the understanding and education of microbes. And he has a book out, and I've talked to a lot of people that like the book, and so I'm just kind of sharing that information. I have no affiliation with him. And I want to say also that Jeff's background is he's not a microbiologist. He's not necessarily an academic. He was a lawyer. And I think they live in Alaska, but him and his wife geek out on this stuff and ended up writing some really good books, and all his experiences from research that he's done. And so kudos to him. It's something that's really taken off. And if you'll give me a second, another guy came in town and spoke, and Scotty with
David Montgomery.
Thank you so much, David. Sorry about that. David Montgomery's the other guy that he's a geologist. He's not necessarily a biologist, but his wife's a biologist, and they're really geeking out on the same exact messaging. And so it was fun to hear both of those speakers, and they have a very consistent message. And it's nothing different than I would tell you. And so they're giving you a really concise, clear, consistent message on how microbes function, etc. Scotty with Duke Gross show, Duke Gross crew, and he's actually the manufacturer of Recharge, had him come out and give a talk to us here in Fort Collins to some growers at New Belgium Brewery just the other day. So thank you, Scotty, for doing that. Scotty also promotes a lot of these guys wherever he can. So we're all about promoting the microbes and using microbes in cultivation.
That talk is actually on YouTube. It was live streamed, and it should be on the Dude Grows channel.
Thank you for saying that it was. I was in the audience with my son that Sunday afternoon. But I don't think there's a picture of me there. I don't think there's one.
Yeah,
We won't demand evidence. That's okay. We
No
believe it.
evidence. Yeah, I was behind the camera.
It's okay.
Thanks.
If you're looking for other resources, another person you can check out is Dr. Elaine Ingram, who has the Soil Food Web. She has an online class. She has free videos on YouTube, which are basically the same content that there's some more advanced stuff in her more recent classes. But if you're looking for a great place to start watching those, I believe it's called the Soil Food Web, and the whole series of videos is on YouTube. And there's maybe like 6 parts or so. And she does a great job, I feel like, of really introducing people and going even into some advanced concepts relatively soon for somebody who's looking to transition to a more, microbial life in the soil based growing methods.
That probably helps for advanced growers too, the fact that she gets quickly into some kind of advanced type stuff, so it doesn't bore the advanced user or the big grower. All right, Steve, I see you wanted to say something. I'm sorry.
No, that's fine, brother. The other resource I was going to recommend is there's another really good website called microbeorganics.org. It's a guy wrote it, I don't know, years ago. It's written in Front Page or Dreamweaver or just raw HTML. It's pretty dated. It's like I don't know. It's like going back in time on a website, but it's all his information on one page. So you can just keep scrolling down. It goes all into how to identify different microbes under a microscope. So if you're doing compost teas and you want to identify what it is that you're looking at, he lists a bunch of different microbes by by genus, so that you can help, identify what it is that you're looking at, and then, do counts so that okay, I added this different brew, and what, and did I hit my target? Does it need long? Helps you fine tune, a 400x microscope, and a little bit of knowledge of the reference sheet like that can really help you make a huge difference when you're really trying to maximize yields.
I think about this some too. I'm actually, I'm a PhD in soil microbiology, and I taught the microbiology classes in graduate school for years in the labs. And there's a lot of really simple techniques that are fun to help quantify microbes. And if that's of value, we should put together some platform, and we could have, we could probably make some videos to show people how to do that. We do it all the time here, and it's just, they're fun exercises for sure. They're like little lab exercises. And if that's of interest, our team's more than willing to start putting together some of those little videos, maybe 2-minute videos, just to show people how to do that. Let me know, because we'll put some resources towards doing that, because again, what we think we do more than anything else, what our job is, to educate and support. And so wherever there's information gaps or desires to learn more, if we can help, we will, for sure.
I would definitely, as not a Colorado grower, love to see the production facility that you guys use. Robots are awesome.
Oh, that's in the R&D, but man, I'll give you a tour of that anytime for sure.
Oh, wow.
Yeah, those videos would be great, especially for, people that are really trying to fine tune. That would be killer, especially because you guys are extremely qualified, to put that kind of stuff out. You are kind of one of the authorities on it. That would be really awesome content. I'm sure it gets you lots of views too.
Yeah, thanks for that feedback, for sure. Yeah.
Hell yeah.
And I have some platforms that I'd love to host your content on. I have no problem hosting that.
Awesome.
I got a whole network been building, and having some problems right now, but we're almost there, and
awesome.
It'd be perfect, because what I want to do is I want to sell your product, but I want to also teach people about whatever you can. If you got all this content to teach people, because that's what I do for a living for the last 12 years. I taught people how to grow cannabis. So I specialize, especially in medical marijuana growers, way more than I do commercial or, punk stoners. Sorry, all you punks out there. But I've really been, I'm 60 years old. I'm all about I teach 45 to 65-year-old med patients how to grow pot. I'm freaking happy. I couldn't be more rewarded. We had a guy, we have a Bud of the Month contest, and the guy won first grow. We taught him how to grow. He bought our nutrients. He bought our genetics. He grew, and he won the Bud of the Month contest, his first grow.
That's awesome.
Against guys that have been growing for 10 or 15 or 20 years, But so it's all about sharing information, and that's why we're all here tonight, right? That's what this whole show is about. I learned so much every week, and I'm a commercial ag grower and hydroponic agricultural grower, and then that came out of learning how to grow cannabis hydroponically, and then I got an offer for a greenhouse. But what you're doing is great. Fantastic. And if you're willing to share that information so readily, that is going to be something that we can throw out there, and I think it'll just boost the whole industry and your company and product exponentially that way, by networking with people like us. That's good for all of us.
Heck yeah. Let's say one other thing too. I was just in Oregon and in Eugene. I mentioned that maybe before the show, Steve and I were talking about that. And I met so many awesome people. And where I learned more than anything else is hands-on and in the field type of thing, talking to people, seeing stuff. And I met like 20 wonderful farmers that are going to have me out to visit their farm when I'm back in Oregon in another month. And if I'm out and about and anyone wants to give me a farm tour or get me out there, I would love to visit your farm. I'll just say that.
You gotta come to San Francisco here. I'm moving up. Well, I'll be talking about that later in the show, but I'll be telling you guys about this. But I got a 200 by 40 greenhouse we're gonna be setting up for just my personal medical.
Awesome.
All right.
That's exactly what I'm talking about.
Over at I'll give you a tour of Ouroboros, because I know they're interested in testing your stuff out on a commercial scale, aquaponics as well. So.
Sweet. Thank you, Steve.
Yeah. Well, do you guys have any other questions? If not, oh, I had one last question for you. I don't want to hold you too much longer. What have you tested a lot with different ethanols and sugars as far as which ones benefit hydro, microbes the most? I guess organic hydro microbes. Is there any ones that are particularly more beneficial than others? And then have you tested anything or worked at all with any ethanol dosing? And then can you talk on that at all?
Man, I'll say I cannot talk on that at all. I don't I don't really understand that very well. I know that I don't necessarily feed our microbes extra sugars. I get the value in that, but what I will say is that what I understand very well is that the plants do feed microbe sugars within the rhizosphere. They actually fix CO2 to put on all their biomass, but in many cases, over 50% of the CO2, carbon dioxide that they fix from the atmosphere, they translocate all the way through and squirt out of their roots as simple sugars, and they've evolved to do that to feed microbes. And so I really like to facilitate that natural plant exudate or plant priming process to feed the microbes once you get them delivered into that rhizosphere or the root zone. And so that's what I would support, but that's where my PhD academic hat for sure. I think there's a lot of folks that use different sugars, different molasses, etc. And I've never experimented with that, and so I don't have any personal experience. I think I would prefer getting it to the plants, getting the microbes to the plants, and letting them exchange that way, carbon for nutrients. Does that make sense?
Yeah, absolutely.
Cool.
Yeah, no, that was a great explanation. Does anyone else have any other questions? If not, we can let Colin go. If
I
he
just
wants
had
to.
one more real quick question. Sorry. Is there any type of nutrients that you recommend using along with your product, or is it as simple as use this product and if there's phosphorus there, it will be found?
Yeah, so I think the latter for sure. Growers using Mammoth P have used all sorts of different nutrients, and we don't see that there's one nutrient line that our product prefers for sure. You'll hear a lot of the growers, like the nectar growers and more the organic growers, that really rave about using Mammoth and using SLF, and SLF's a great product also. It's an enzyme product, a microbial derived product for sure that works very well. But I think in the eyes of the microbes and plants, to a large degree, nutrients are nutrients, and the microbes in Mammoth P and other microbial products will just break down those nutrients and make sure that they're bioavailable so the plants can take them up. And so I don't think in most cases the nutrient line should matter as much for the microbial products.
Awesome.
Got it.
Well, that's my last question, guys.
All right. Any other questions? Hit me up. Open door policy over here at Crescentia.
Did you post? Did we get the link to this posted in the chat? I'm not asking you, Steve.
Which chat?
The link to Mammoth P.
Yeah. So the link to Mammoth P and all their different stuff is in the description as always. All of
Okay.
our guest stuff is always in the description. I handle it all ahead of time. For anyone that's ever interested, if we ever have a guest on the show, their company information or contact information is always available in the description. Do you want to tell people how to find you and how to reach out to you and how they can sign up their local hydro store for trying to carry your products and other things?
Absolutely. Thanks a lot. So you can always visit us at mammothmicrobes.com. That's our website. You can always email info@growcentia.com. Mo, who handles a lot of the communications specialist, is her title, is mo@growcentia.com. And my email is Colin with one L, COLN @growcentia.com. Again, feel free to email. I'll respond to you for sure, and we can interact on a phone call or a text or an email, whatever. I think that's it. And tell your grocery stores to carry Mammoth P. And we'll make sure we get it there for you.
Well, you just opened that door really wide, didn't you?
Yeah, I know. It's okay.
No, the stuff's really good and it's worth using, and you can't. It's almost impossible to kill your fish with it. I've tried. So we actually do. When I used to work, we actually used to do a lethality test, and we put goldfish in a 10-gallon aquarium, a couple of feeder fish that we've always kept a couple bunch on hand, and we take a couple of them, throw in a 10-gallon tank, and then do like 40 times the dosage with something and just see what it would do, because I have customers that'll do that. I came up with, or developed the product Aqua KH Up, or I think it's called. It's sold by the aquaponics source, and we had a customer overdose it by like 40 times, and he still didn't kill a fish. I mean, his plants were jacked up, but he didn't kill anything, and that's the important part. But like the idea is you have to plan for like just someone that has never done this before and might have a completely different viewpoint as to how they think this should work.
Yeah, that's right.
So, but thanks a lot for coming on the show. You're welcome to hang out for the rest of the show, or I know it's getting kind of late over where you are. You probably want to get running, but you're welcome to stay for the rest, or if not, thank you so much for joining us. We've been trying to get you on the show for a long time. We very much appreciate you taking the time to join us, and it's been a huge pleasure. Thank you so much.
Thanks, Steven. Thanks, everyone else. I really appreciate it. I'll be happy to come back anytime. I'm going to bounce, get home and take care of some things, but have a great night, everyone.
Incredible.
Yeah, thanks for answering all the questions.
Yeah, absolutely.
And expect an email. I'm just saying.
Expect 1 or 2.
2 or 3 or 4. Yeah.
Hey, have a good night, everyone.
Yep,
Thanks.
thank you.
Thank you.
Guys, Jared had to run. He had a last-minute emergency come up. He's going to try and join us next week. He was on the show earlier, and he just had something come up while he was on the show and had to leave us. He might join us again sometime before 9. If he's able to make it, he's going to come back and do a segment. If not, we're just going to schedule him on. We do apologize. Colin's section ended up running kind of long, but I think we were all super excited to have him on finally. So
What a great show. Yeah,
Yeah, we were grilling him pretty good.
What are you guys? What are you guys up? What have you guys been up to this week? Before I go on about what I've been doing, and then we can talk about this Jeff Sessions thing this week.
I'll go
Who wants
last.
to go first?
What's that?
Who wants to go first with what they've been doing this week?
So I guess I'll go first.
That's
We
easy.
must be patient. I don't know.
I guess, well, let's see. For the past couple days, I've been working on the screens that I made myself for Dry Sift. And I'm definitely looking forward to using those because I was able to harvest my buddy's plants on Monday, and then I harvested a few of mine today. So I'm looking forward to making a lot of fucking hash with that and making some videos of that and whatnot. Making some melty hash videos and whatnot. But other than that, that's pretty much it. That's the highlight of what we do. Harvest time.
It's all about the harvest.
That girl with the LEDs that was supposed to reveal this week. When did this week? Who was that?
That was Fish Ganja guy, but we're not going to do that. We're not going to do that. So next week, we're not going to do that yet. There's a plan for it. So next week, if all goes well, assuming there's no hiccups, we're going to have Black Dog LED on. So we will then have both of the companies on whose lights are in the competition. And then in the episode after that, or whenever he gets his lab testing, whichever one comes first. If he doesn't get his lab testing in time, we'll wait a week or two. And then
I've got a lot of members on the forum that want to hear those guys in the LED companies talk about the cob lights.
I can PM you and tell you which one did better, but I can't tell the public because I'm not to.
Right.
But I know.
Just have both of the guys on the same show.
The other
have them box it out, and then whoever loses has to pull their market from.
We won't tell Fish Ganja guy that we're gonna do that, and then we'll be like, hold on a second, and then we'll have them all join, and they'll be like, all right, now do the results. That would be too funny. No, that would be mean. It would be hysterical though.
Hysterically mean.
Hysterically mean. It's okay. We all love Fish Ganja guy.
Well,
Definitely have some rage quitting on that one.
Yeah.
so you can tell the truth, which lab system did better.
Oh. Anyways, it'll be fun to have both LED companies. I use the Black Dogs a lot back in the aquaponics source. We used to carry them. I will say they're extremely reliable. We only ever had one ever go wrong, and the only thing that went wrong with it was the temperature sensor for the fan when the fan would kick in. So it was like automatically turning itself off to make sure it was preserving itself. And they had it back to us in like an hour. We dropped it off at the place, and the guy dropped it back off at the shop. Took us. I mean, they're awesome. Their customer service is super kick-ass. So. But yeah, they'll
You
be
said
on
that was the
next
Black
week.
Dog?
Yep, Black Dog. They'll be on next week. And then I also got a chance to see
Sweet.
up at Ouroboros. They just got a couple of Spectrum Kings. They have a couple of Mother's Little Helpers in their veg room. They have like a little veg seed start room. I call it the veg room. It's a seed start room. Or nursery.
Propagation room.
What did you say? Fornication room?
Propagation.
Oh, propagation. Plant. That's what we should put above the door. Plant fornication room. Yeah.
That's the breeding.
Of like a sunflower bending over another sunflower or something. It'd be funny.
Oh man, this is fun. All right, so what's next?
I think it's Marty's next.
Oh, well, let's see. I haven't been doing too much. I picked up a cut of pre-98 Bubba that I'm going to throw in the start vegging. I haven't really done too much else besides that. I've been running around prepping stuff to get ready to try and sell my house and get out of this fucking county. So,
how's the baby?
Good. Obviously, spend a lot of time doing that. Probably most of my time doing that. So,
Yeah.
not much gardening happening other than here and there. But
Okay, I ordered a plant.
yeah. And so getting the garden reloaded and trying to get some videos shot. And yeah, really not too much else going on. I went out and toured a rec grow that's in not good shape. I mean, they don't have their plants in the ground yet or anything. And so just kind of went through and just kind of took a tour of that. And it just goes to show you that, and it's not, funding is important, but it's not, it's not going to fill pots or build fences or any of those things. It's, those guys spend a lot of money out there and aren't really in a position to grow anything. And, it's one thing to have like a giant orchard sprayer for foliar spraying all of the plants that you're supposed to have. But if you don't even have plants, your orchard sprayer is not a great investment.
I took a $40,000 slab a couple of weeks ago. They wanted me to design a greenhouse, aquaponics greenhouse too, and I talked to Steve about it. They only spent, they had a $100,000 budget, and the first thing they did was build a $40,000 slab with no radiant heating or anything in it. So they got a big piece of rock, and 40% of their money is gone. Sorry, I had to throw that in. It was like, it seemed like a
no, and it's it's tough, and then they're
This is why you hire a consultant.
Yeah, exactly. They said they don't need one yet.
Well, a lot of times the people that really need consultants don't have enough money left to pay a consultant. So, that's kind of a tough situation. Like when, people end up having to depend on the next harvest to be able to pay somebody or be able to pay employees or any of that. It's just the green rush as they call it. It's got a wide spectrum of people taking a stab at it, I suppose. And they still have a lot of resources and can get a lot done, and I'm sure they will. Just I don't know about this season, being that it's it's mid-June and they have a third of the plants they need, and their greenhouses are put together incorrectly, and there's a number of different things that have to get done. So it's really it's interesting to see, because I've been to a number of them that are obviously well put together. But maybe underfunded, for instance. A lot of medical growers, for instance, that were doing it for a while and developed grows over time. may not be the most well-funded grows, but are extremely productive for the money that they put in. And so to see the opposite spectrum of somebody putting in a bunch of money to literally not get anything out of it yet, was really sort of eye-opening for me. Like, it's, it really is still a business that you have to be smart about. And, that's sort of was my eye-opening experience of the week.
They try to go too big too fast. Instead of learning how to set it up and get it done and then expand, everybody, like this particular customer I was talking about, they wanted 20,000. They wanted to start 20. Their plan was to start 20,000 seeds, which of course, we all know that would be just stupid, because if you want 20,000 plants, you don't start 20,000 seeds. But they wouldn't. They didn't want to hear it. I tried to tell them about setting up a clone operation and all that stuff. Buy 100 seeds.
20,000 20,000 seeds.
Yeah, can you imagine?
Yeah, I know. Yeah, have you seen my Jamaican video? My Jamaican videos. It's a field with thousands of different phenomes. Thousands of fucking phenomes.
Field in Jamaica. Yeah, this guy's trying to build a building.
I know, but you can't. The problem is, and let's hold on. Let's extrapolate on this, because let's explain why this is stupid. Someone, I don't know, for real, like, because someone out there might be like, well, if you plant them all, isn't that gonna be the fastest way to harvest? Like, no. So this is the problem with this. Every seed can be a different phenome type. The phenome type is the genome combined with the environment. Each plant, each set of genetics, and each seed will act differently. The reason why you use moms and clones is so that you can get even, consistent production, so that when you go back 3 months later or 9 months later or 2 years later, it's the same cut, it's the same strain, it's the same, genetics, it's the same potency. The plants will be, same predictable harvest schedule. They'll have, pretty damn similar potency, as long as your nutrient input's the same, and it just makes for a good, consistent product. That's what you need to achieve when you're growing commercial cannabis. They don't care if you have a ton of it. If I'm going to have like a pound of everything, I don't want 1,500 pounds of different stuff. I need, 200 pounds of one and 300 pounds of another and 400 pounds of another. That's what they want. So that's why that's important.
Yep. Very good. And, and by doing clones, you get, it's all, all the plants are basically close to being the same size. That's one of the things that's real important about that's why you do clones and put them in a big giant place, because they'll be the same size, genetically pretty much the same. They're going to be the same as the mother. Yeah, they're going to be very close. The height, everything is going to be the same. So once you figure out all your spacing and all, and how much you want to do, you can just keep going and going and going. You don't need 20,000 plants tomorrow. You need, like you pick out your like you said, you find out what you need genetically, and then you appropriate spaces in your building for that. Because these guys wanted to do a total like environmental control, and I said, you got $100,000 budget, you spent $40,000 on a slab. How are you going to control that? You can build a building next. What are you going to be like? Have 10,000 left for environmental control? I mean,
here's the other one. Here's the other one people don't. So there's two things there. So one of the other things people don't plan for is paying anybody to do it. They'll just spend all of it in equipment costs, or spend all of it in permits and equipment costs, and don't set aside any money to pay anyone to work on it. If someone has to still spend labor hours on it and pay their bills, which is something else I've seen, but not, with at least initial proposals.
Right. And as an owner, if you don't plan to pay somebody to do it, then it's you. Like, that's what it comes down to. And those are really the only two options. There isn't another one. And I do feel like that's a mistake that a lot of people make. And consistency obviously being what you need to have in terms of plants is important, but plants at all is a start. When you're supposed to be able to have like 900 plants and you only have 300, something went wrong somewhere. And I feel like people just don't realize how much planning comes into it, because that is going to take time to do it correctly. It's not going to be like, like you said, oh yeah, that's the fastest way to get that many plants. You just throw seeds in the ground and grow and harvest. Well, okay, yes, you'll have plants. We talked about the inconsistencies. So when you do it right and you want to, select your phenomes and be able to do that, that's going to take at least a few months worth of work just to select those. If you're doing it based off of flower, for instance, if you're doing it just based off of traits that happen in veg, then that's maybe not as informed. So if you really want to do that, that full process, it's going to take months just to grow out, flower, select, and have other ones growing up during that same time. So you're talking at least months to ramp up to where you need to be at, as opposed to just saying, okay, well, as soon as the greenhouses are going to be done, I'm going to throw these plants in and I'm going to harvest immediately as soon as they're done. And that's what people plan to do. And then, I think they plan to have optimal results, and then when they cut corners, they end up with not optimal yields, and then that causes issues for their cash flow. And again, it's just like any other business. If you plan to sell your strawberries for 5 times as much money as you can sell your strawberries for, you're going to have business problems. In the same way, if you plan to sell your cannabis for 5 times as much as you can actually get for it, then that's going to be a problem. And just seeing that in some of the local rec businesses around here, for sure.
And with that in mind, what's the going rate for pounds up there right now?
Well, outdoor has been pretty consistent. For quality, it's held pretty strong at like 1,200. I would say for average quality, I would say 1,000 would probably be about that. And then indoor is usually about 2, and it holds pretty steady at that, just because people understand you have higher costs from growing it. Some people say it's higher quality and other stuff, but I think everybody agrees that you have to pay for electricity to power lights to grow indoors. So it costs more, therefore.
And you said 2?
It costs more. What's that?
Marty, you said 2?
Yeah, 2K.
2? Oh.
For indoor.
Nice. Yeah. Yeah, I'm at, we're near that here in our area, near that, just a little below that, really. I would say we're over here, we're at like maybe probably 15 or 16 per pound, or maybe 12 for mids, or not mids, but 12 for pretty good grade, but not like blow your skirt up. Tastes good, like you know what it is when it comes to money. To me, not my personal choice. I want something that I get an effect out of, but what the desired effect. But a lot of people, they buy that taste. And the taste is going to 20 a gram. It's going 120 a quarter over here. So you're going 400 an ounce or so, something like that. And so where's that put us at a pound? 48. So wait a minute. 400 an ounce.
You got to factor in wholesale and all.
Well, and also the thing about taste, though, is that whenever you're talking about terpenes, like that's what modulates the high. So, I mean, if you compare it to like the synthetic THC that was made in the form of a pill, I think they call it Marinol or something like that.
Yeah.
Whenever you take that, a lot of people just reported having a lot of anxiety and whatnot. And then whenever you add the different terpene ratios in with your cannabinoids, then you start to feel different effects, because as we all know, most cannabis that people want to smoke is going to have a pretty decent amount of THC, but especially for me, like I want it to have a real good taste, and like I said, it always seems that if whatever particular strain that I'm smoking or whatever has a higher terpene profile, then I'm getting much more effects from it.
Can I ask something? When you brought the anxiety angle, can you guys answer this for me? Because I'm not clarified here. You're talking about the terpenes, and then you were talking about the anxiety, though. So when you first said when you gave you the anxiety, I was thinking, is that because it was harvested possible? I mean, because was this made by a corporation where possibly sativa was harvested early and it wasn't quality?
No. Molecularly, it's the same as THC. So you're basically just talking about a pure THC extract, completely devoid of any other types of oils or terpenes or cannabinoids that would be going along with it if it was growing naturally.
Okay.
The other thing I was going to mention is there's a, I don't know if you guys saw this a couple months ago. They did a synthetic cannabinoid test, and was it Spain or Portugal? Do you remember, Marty? The one that went really bad? And it killed a bunch of people. They did, I think it was 12 people they tested it on, or 15 people they tested it on, and like 9 of them died within the first 24 to 48 hours.
Yeah, I don't remember either.
Horribly wrong. I know we talked about it on the show, I thought.
Yeah, I can't remember where it was at though.
It was in Europe. I think it was Spain or Portugal. It was a synthetic cannabinoid replacement.
Yeah, well, that's the problem with synthetics.
Yeah, for sure.
I've had years ago when I was running the Grow Report for Xandor out of California, and we were our servers and website were based in Amsterdam. There was a bunch of scares in England and all with stuff like that. I'm not going to go long into that, but that and the tainted with the glass, it was really weird. That was some of the stuff that goes on that you learn about and hear about. But yeah, right along the same lines is what you're talking about. You know, it's been going on for years and years, but synthetic always seemed to be the one that caused a lot of people problems. When through the years I've read, we don't do as much as where I'm at now. And I love growing marijuana, but at the Grow Report they allowed all kind of psychotropics and everything from talking about acids, mushrooms, everything. It was all psychedelics, everything. And so I picked up a little bit here because again, I've been loving podcasts for a while, and we were a podcast network, so it's pretty cool. I got to crazy shit, but yeah, am I wrong here? Aren't the synthetics way more dangerous and unstable?
What was that last part?
A lot of the times, the synthetic cannabinoids are like much bigger than anything that you'd find in a cannabis plant, So like they cause so many times more of an effect than cannabis could ever possibly cause. So like in a sense, as far as cannabis goes, there's like a natural buffer. There's like a natural safe limit buffer in cannabis, and the stuff that they make synthetically is just like so many more times bigger, so it'll have so much more of an effect.
And it just keeps going and going and going and going and going.
Yeah, so I found an article here. It was in France. So it was in France, and they had a total of 90 people had taken at least some dosage of the drug. Of those, 49 of them became ill, and a couple of them became completely brain dead. It was an experimental painkiller using synthetic cannabinoids.
Wow. I wonder what kind of dosage they were talking about.
Yeah.
Well, it doesn't matter. It was synthetic.
Or even how it was administered. Yeah, I mean, either way, regardless, it's synthetics, and there's no need for it.
Synthetics. That's my opinion, but everybody has their own way to go.
Awesome. Alrighty. Anything else that you're working on? Oh, baby duties. Okay. Marty had to run. Check out his channel, AP Meds.
I'll be right back.
Oh, he's gonna leave us now too, y'all motherfuckers.
No, I'll be right back.
Well, I'm gonna wait for you to cover it. I guess I'll just talk about what I've been up to. I'm in the process of relocating to the Bay Area. I'm working on multiple projects now, both cannabis and non-cannabis related up there, which I'll have some info for you guys soon on. I also now worked out, as soon as I get up there anyway, I got pictures of it. I'll have to do a video of this new greenhouse. Well, it's not new, it's older, but it'll be a 200 by 40 greenhouse that we'll be fixing up to do aquaponic cannabis production for my personal medical grow along with my roommates. So that'll be great. And one other friend of ours, the three of us, to grow in a 200 by 40. So that'll be really awesome. The location is really killer. and it'll be right on the right outside the Bay Area. So it'll be really dope. I'll have more info on that soon. Working on a bunch of cool stuff right now that I'll be able to talk more about once we have it a little more together and able to package into a label for you guys, or brand label for you guys. What else can I talk about? Oh, Puerto Rico has got a new ridiculous law where they ban flower sales, which is totally ridiculous. Like, why would you allow cannabis growing in a medical state and only allow concentrates and edibles? It's completely ridiculous. We've seen this happen in a couple of different states. So they've totally jacked up the market, and well, they're still on schedule to open. They're definitely delayed now, so that's definitely screwed up some of the projects that I've been working on down there. It's getting really old, I'm not gonna lie, where you get a cannabis company, you have tons of money poured into it, hundreds of thousands of dollars, permits, licensing, zoning, land permits, construction permits, all this crap, and then they randomly change one law on a whim and screw you over completely.
Yeah.
It's why you need to have a lack of local control on any of these cannabis bills. All it does is hurt the industry and hurt people that invest, hurts investors more than anyone else. So make sure that you rail against them whenever you have the option to, in any kind of setting where they're proposing new laws.
That's the truth. It's ridiculous that a state passes a law and then they give these small, all the communities the right to use it or abuse it. And you get the people, like you said, they invest, and it's actually legal for a while, and then there's a different mayor or a different city council.
It almost puts people back in the situation of the federal illegality, because then you have every county that can decide for themselves whether they actually want it there or not, so if you don't have protection against local entities, then you really don't have shit.
You're gonna end up with a local religious Reich, and they can ban your stuff for whatever ridiculous bullshit ass reason they want to pull out of their ass, or some other group that is completely stuck in the 1940s.
But that could work out too. The people that want to be in that place can move there, and the people that want cannabis, they can move to the other county. And
no, but the point is, I'm a taxpayer, right? So if I'm a taxpayer and I can buy alcohol super easily from, I can go to 7-Eleven, a gas station, 24 hours a day and get alcohol. Maybe not 24 hours a day, but California, what is it? Up until 2 a.m. or something, you can buy booze at the gas station. You can't tell me that. Not only that, it should be forced on them so that their schools get more funding, so that the townships have more money for homeless thing. Look at Aurora, Colorado, where they're using it to fund the homeless shelter. Like, this should be forced on them because it helps their community far more than any negativity. And on top of that, we talked about this on the show earlier in the week, where they've done studies where when they close down dispensaries, there's direct correlation with an uptick in crime because of the petty drug sales. So all it does is create more crime and defunds schools, prevents funding going to schools and hurts our communities. So they shouldn't even have the option. This shouldn't even be up for debate. There's 2 dry counties left in the entire country. We got past that bullshit. This should be in the same realm.
Yeah, it's always great to fire your ass up.
The other one they need to stop fucking doing is banning production in unincorporated areas of the county. These are the areas that need economic influx and communities that need the money. They have a huge install of people that need jobs that have lots of time on their hands, and a lot of these guys are really skilled workers that live out. I'm just thinking of California and the desert and some of the mountains areas and stuff like that. There's a lot of people out there that need jobs that are hard workers, and all it does is screw over the people that need it the most economically. That was random.
Yeah, exactly. And the example I can always think about from that is Texas. with the CBD law that's fixing to go into effect, they'll have a fucking monopoly in Austin, Texas, you know. So you have one city in the entire state of Texas to get cannabis from. So how much sense does that fucking make?
Yeah, it's just fucking asinine. And not only that, what if you're a cancer patient who lives in, you know, Brownsville or Galveston or, you know, somewhere else in the state? You know, what they're supposed to drive, what, 8 hours to go buy their medicine once
Yeah,
a
you
week? Like,
better
fuck
fill up
you.
that gas tank, be ready
Yeah,
for a vacation or some
are they gonna
shit.
get tax credits to reimburse them for their gas they
Yeah,
spend?
seriously, that's, I mean, that's an
This is,
absolutely
I
logical
want to,
question,
you know
you
what I
know?
want to see then? I want to see opioids for one distribution center per state. That's what I want to see.
That's the thing. Chicken Hole has to drive 9 hours to get to Colorado to get his meds if he wants to get meds because he lives out there.
Yeah,
Allegedly.
exactly.
Allegedly.
Allegedly.
One word makes everything better. Um,
He allegedly drives. Yeah.
allegedly. So yeah, so that'll be that's that. So let's talk on the topic of the week that I want to touch on is this Jeff Sessions bullshit that happened the last couple of days that we didn't have a chance to talk about it on the show, I don't think, on Monday because it's happened since then. Jeff Sessions wrote a letter to Congress asking them to stop the renewal of a bill that prevents federal funds from going after medical cannabis companies abiding by state law and medical cannabis patients, including cancer patients, AIDS patients, people with seizure disorders.
Shay's trying to fill up those prisons for the ones that fund his campaign. Hell,
The KKK Keebler elf needs to piss off and, you know, stop trying to screw up an industry that is generating billions of dollars and saving millions of lives per year. You know, we have to stand up to this shit. We cannot tolerate it. If he does start cracking down on raids, we need to make his life hell. We need to make sure every restaurant he goes to, every time he ever goes out, he's immediately greeted with angry people that are pissed off at him for taking away the medicine from their children, from their wives, from their husbands, from their parents, from everyone else that needs this as a medicine. This is bullshit. We fought far too long and far too hard to let this, you know, racist elf come in and take it all away. It's bullshit.
yeah, it completely goes against Donald Trump's campaign promise to leave the fucking states alone that have already legalized it and have some type of legislature in place.
Well,
Yep.
did he really promise that? He promised everything
He wrote
on
a-
both sides.
He wrote a letter. He wrote a formal letter to Congress on Tuesday that asked them to allow him to go after medical growers and medical patients specifically. In fact, I could probably pull the letter up here. Give me 2 seconds.
So are all you guys happy out there now? Are you happy now? You think you're going to be happy
And
now?
that's actually kind of scary because the way things are going right now, like I don't know, it really could go either way, and it's a
Well,
scary thing to think about.
I don't see him surviving. There's no way. He's got a week, if that. Like, I'll be shocked if he's not out in a week. He's already threatened to resign once.
Well, he was covering Donald Trump's ass with his testimony, apparently refusing to answer questions about their conversations. So, I don't know.
This is his exact quote. I believe it would be unwise for Congress to restrict the discretion of the department to fund particular prosecutions, particularly in the midst of a drug epidemic and potential long-term uptick in violent crime. Let's pick apart this bullshit fucking statement, okay? First of all, crime nationwide is at an all-time low. You can look at that statistic on the FBI's website. That's a fact. Anyone can Google this. Okay, bullshit number 2. Historic drug epidemic. Yes, thanks to who? The pharmaceutical companies and their opioid addiction. And then when people get off the opioids and they can't get the pills anymore, they switch to heroin because it's the same shit. You know, Oxycontin is the same molecule as heroin. It's one little atom off.
Right.
You know, so don't fool yourself. You know, Adderall, the same shit that everyone's giving their kids, is the same as meth. It's one molecule difference.
Meth.
Right.
Yep.
So the next time you give little Jimmy
All that matters is the dosage,
ADHD
people.
medication, remember that you're giving him meth. So
The difference between poison and medicine is in the dose.
it's on the label. So, I mean, this is completely ridiculous. And not only that, we touched on this in the show on Monday. If you guys aren't aware, we do a Monday show, totally news-based, called Last Week in Cannabis, where we touch on a bunch of different news topics, anywhere from 20 to 50 news topics, depending on the week. The first week, we had a lot more news than we did last week. So, but we're going to keep going with that, and we've had a blast. I'm learning a lot. It's been really good for staying current with the markets and everything. It's been really fun. So definitely check it out. But we were talking about this, how there was a study done where they showed that states that have legal medical marijuana had a 40% decrease in opioid deaths because people were using the cannabis over the opioids as a painkiller, or they were using it in conjunction with the painkiller, and they weren't overdosing because they weren't taking as much. It also helps with what was it, Arizona? The Arizona state medical board is using cannabis to treat people's withdrawal from opioids. So when you have people involved in government using it to treat people's opioids withdrawals, this is stupid. There's no more arguing about this. This guy's, and you know why? And I'll tell you why. You know why Jeff Sessions is against this? You know who came out and said that it was pushing for medical diversity? Because Al Sharpton's for it this week. So, the racist elf over there has to be against anything that's for any kind of minority, increasing their financial gain. That's a big reason why a lot of these people are against it.
On board. Sharpton's on board. Sorry, Steve. Sharpton's on board with this for the poor and for the elderly and the habit, and then the government official is okay. Yeah, okay. I see the writing on the wall. Okay.
Well, so, he's just racist. So anytime that, and that's what a lot of this stuff is. A lot of these laws are where they're preventing people with previous convictions. It's not about making for good market, preventing good. It's about continued racial oppression and making sure that continued cultures and area, social groups that have been oppressed for a long time are continually, continue to be oppressed and don't receive the financial gain from the shit that they're good at, that they've been doing for a long time. That's why you don't see a lot of minority groups represented in cannabis ownership.
Wow. I never looked at it like that. I figured they'd have their own foothold. Minorities they got money.
Well, they do in some places, but
that's one view that I have always seen from the start. Is how the minorities, which are the people that have suffered under these laws for so many years,
the most
are being left out of this, left out of the revenue that's being generated and all that.
Yeah,
absolutely.
It's just completely ironic.
Probably racially profiled, and so somebody you're talking about when you were talking about the first offense, second offense, a lot of that might have been not that. Hey, okay, law's a law. Whenever I've gotten caught or busted, it's all about. It's about hey, you broke the law, you got caught, so you just be a gentleman and deal with it, and then fight on to live another day. So, well, it's sad, but a lot of these people, when we're talking about first offense, when they're facing stiffer crimes or incarceration because all they did was have some pot with all the other shit going on out there, they had some pot, they didn't have meth, they didn't have coke. They didn't shoot anybody, they didn't knife anybody, they didn't rob anybody. They weren't even smoking cigarettes. They just had some pot. Oh, that's
the way it goes.
And like the cocaine laws. Look at coke versus crack. Cocaine, a stronger drug, will get used to get you 6 months if you're a white guy, or even probation. If you're a black person with crack, that equaled half the amount of actual active ingredient as the cocaine, or less. You'd end up with 8 years, 12 years. And again, it's just about racial oppression. Not even just racial, oppressing certain social groups. You know?
Well, some of that average is skewed though by the mandatory sentences, though. So
Yeah.
that's
Well,
it's a little skewed because of mandatory, because there was more than one conviction. So but I agree with you 100%. Minorities were getting. We all know that if your brain is open, we all know.
It's simply because it's easier to get a conviction on the minorities. You know, those are the ones that they target because we don't have money to fight the system. You know, like everybody else. You take a rich kid that lives over on the rich side of town that has a well-connected daddy or whatever with some money. As soon as they get an attorney, you know, they're likely to drop the case or whatever. They don't want to fight against that.
What's the point? White boys ain't standing out on the corner. They're not living in the hood where the cops are running down there and deciding he looks suspicious. Even if he's just carrying a little bit of weed to go home and smoke it, he gets busted because he was walking down the street and he was the wrong color.
Yep. Or you get a DWB, driving while black or brown, whatever you want to call it.
Well, I've been arrested. I've been arrested for being white in a black neighborhood too. So I don't think.
I've been there.
Yeah. So, and I gave them hell, man. I made a show in the video. I don't even know if they may show that shit for training. I don't know, but yeah, I made a big deal. I had the black folks cracking the fuck up. But yeah, I was arrested for driving while white in a black neighborhood once, and so I know how it feels. All you brothers, I mean, you know, you probably don't.
Not really.
That's what became my brother. But
I was about to say, I don't know about that.
You know, the closest I felt to that was in Jamaica.
You don't live where I live, Bubba. You don't live where I live. I kind of know. Okay.
In Jamaica, they're kind of not most of Jamaica is great, but there's one little area particularly near Spanish Town where it's very like, I don't know about you. It's like going to the South. It's like the reverse of the South in the 1800s. It's very strange, but
I wish I could come down there when you go down to Jamaica. I want to come down there, man.
I think I got a pretty long list now of people that are coming.
What's talking to boat that goes to Jamaica? So
I'm gonna talk. I'm working on some cool content and we got some cool guests that'll be on for that trip, and we're gonna do a ton of video content. You guys are gonna like who we got that's gonna be going with me. I'm not gonna say yet because I don't want to spoil it, and I want to make sure that like nothing falls through with our plane tickets and stuff. But I got a pretty cool surprise for everybody planning with somebody you guys are already aware of or already follow in the cannabis community.
Yeah. Cool. That will be awesome. Nice surprise. That was a big shot, man.
So I actually had a question from somebody. There was two questions. There was like a Facebook question I want to cover, and then I had another question that got emailed to me. Somebody asked me if I could touch on what to do if you get raided by the police. And I would say it depends on your arrest. So if it's a minor arrest, like you just get caught with a bag of weed or whatever, the biggest thing is make sure that if it was a car search or something like that, that it was a legal search. And, you know, don't just shut the fuck up if you get arrested and just say you want a lawyer. Don't say a word. The other thing I recommend to you, if they come in like Call of Duty style with the flashbang grenades and the whole nine, do not waive your right to a speedy trial. Immediately call for a trial in 10 days, which I think is the fastest that you can do it. It's 10 days or 30 days, something like that. But they have to. It's 10 to 30 days they have to bring the thing to trial. Here's the deal. The police, each one of those officers, the people in the helicopter, if they did that, all the different cops in the different vehicles, the support team, all the different dudes have to give written accounts of what happened and submit it as evidence, and a whole bunch of paperwork and a bunch of bullshit. If you call for an immediate trial, they physically do not have the man hours in 10 days or even 30 days to bring it to trial. They will drop the case because they physically cannot. There just isn't enough manpower because they have other cases they're on that they can devote to solely doing it. So you could have a whole huge grow up way over your plant count, have them come in raid Call of Duty style, call for an immediate trial, and they'll be forced to drop it because if they don't have their charges ready in time for the judge is going to throw out the case. So that's like two of the bigger ways. And granted, it's not gonna make it out every time. It's not gonna help you. But in the majority of cases, those are two good ways. The other thing is if they accuse you of making butane honey oil, unless they catch you like in the act of doing it, they cannot submit it as evidence because they have to prove, and this is something that cops cannot do, cops cannot test for the presence of butane in a cannabis extract. They can say that it's a concentrated form of extract, and then they have to determine, they can only say it's concentrated form of cannabis if you have the plant material present as well. So if you do all of your stuff in one run, there's no plant material present, there's nothing to test it against. So those are ways that you can use to help, get yourself out of it. And at least help them, basically punch holes in the prosecution's arguments as well. But basically the biggest thing is if they come in Call of Duty style, call for an immediate trial and you'll get your case thrown out.
Well, this was actually my topic of the week that I asked you to include in the show tonight. It wasn't from an email. It was my idea. So I got an idea. I was not going to the honey oil, over maximum allowed plants. I was going more for the guy that's at home with 4 or 5 plants, and the cop knocks on your door, and he asks to come in. And first of all, you always say no. Because and the second thing out of your mouth should say, do you have a warrant? If they do not have a warrant, then they're not allowed to come in. They'll threaten you with, we'll just stay here, but they won't, probably.
No, they won't. No, because it says they have to go all the way to a judge, and they have to get them to hear it, and then they got to explain to them why they need to violate your constitutional thing. And especially if they've already fucked up at that point, you can go and testify against their warrant in person, especially if they've already approached your property.
But the thing is, what's real important to know is that if you've been growing indoors and you've been doing rule number 1, 2, and 3 about growing, don't tell anybody.
Yeah, shut the fuck up.
Don't tell anybody. Everybody you look at you should look at like they have a badge on their fucking shirt. Yep. If you never tell anybody, there's nobody to testify in court for a prosecutor to get a warrant so the police can come to your door with a warrant. If you do not tell them, if you tell nobody, you'll want to brag about it. But if you want to be out of jail after years of growing pot, then you do it and you tell nobody. If you're in a state that does not have medical marijuana or recreational marijuana, you need to keep it to yourself. You'll impress your friends when you got the best weed they ever smoked.
Yep. And you tell them you got it from your guy. You got this guy random. You got the dude you met at the bar, and it's just some fucking, you make up like a fake dude, and that's where it comes from. They don't ever need to know that you grow at all.
And there's another thing. When you answer the door to a law enforcement agent, do not step across your threshold. Once you step across your threshold, they can take control of you bodily because you are no longer in your home domain. So make sure you do not allow them to step across your threshold without a warrant, because then they're coming in whether you like it or not. So if you got a warrant, fuck it. You just fuck it and call normal if you want to take the.
I just remember, plain sight doctrine means anything viewable from the front door is admissible as evidence. That's so right. If you have like pots, plants, like in the living room, you open the door and they're visible in the back, he can use that as evidence to come in.
Or you got an ashtray full of roaches. Okay.
Yeah.
Keep your shit where it's mobile. You put it in your little closet all the time when you're not sitting at home all night or chilling out, and then you put it away. Don't leave it on your table where they can look in there. But the first thing is you limit the access to what they can see in plain sight by never letting them cross your threshold of your domain. And you never cross the threshold of your front door onto the porch of your domain or anywhere else, because then they got you. If they want you, they can just fuck with you. They might not arrest you, but they can fuck with you. They cannot fuck with you if you're standing in your house and you don't let them in. Yep. You can open the door. If they take you from inside your door like that without a warrant, well, that's just one of those fruit of the poisonous tree situations. And any decent lawyer for $2 or $3 or $4 grand will, fuck them up. You might not even need a lawyer if you're smart enough, but, hey, when it comes to your home and your pot, and, you might, well, you're not going to get your pot back where I live.
Yeah, not where you live.
No. Anyway, all right. So I'm giving it up. That was my rant for the night. That's what I really wanted to get out to you folks because it was something that happened close to me the other day, like yesterday, and I was talking with Steve, and I said, you know what? We need to talk about that topic because that's a real important topic when we're talking about people growing, and a lot of our members and friends and followers are from states where they do not allow any rights to use a simple herb, as Steve would say, and I won't start saying fuck them and fuck this, but you kick out of, I get him rocking. He gets rocking sometimes. So that's all I got to say, really. I hope you enjoyed my rant. I hope you take it to heart. It was a rant so that you might remember it, because that might keep you from going to jail or losing your stash or whatever. The only other thing I would say is once that all happens, they're gone. Pack that shit up, and however you got to do it, don't be stupid. Don't get caught like they're down the road. But pack that shit up. Make sure and get that shit the hell out of your property.
And remember, cell phones or GPS tracking devices should never be saying anything on the phone, and anytime you're transporting, it should never be in your vehicle.
Right. But you can turn off your GPS.
No, they can turn it back on without you knowing. You don't bring a phone.
See, there you go. There's Big Brother up your ass again, folks.
Yep. They also have things like stingrays that can grab all your information and everything else. Although there is software out there that can automatically kill the battery connection to your phone should you detect that. I have one that's called Spy Cell Catcher, and it's just cool to let people know when they're, illegally driving through neighborhoods, illegally stealing everyone's in the neighborhood's cell phone data, without a warrant, which is illegal. They have to get a warrant for a specific person. That was ruled 2 years ago, because of a FISA court ruling. Anyways, we had another question from the Facebook group. For those of you guys that are just joining us for the first time, I know we have quite a few people, especially with the Mammoth P being on the show today. We had a little bit more regular viewers than normal this week. We have a Facebook group called Aquaponic Cannabis Growers. It's a closed group, but we have lots of really awesome growers and lots of cool topics, good feedback. It's a really great community. It's one of the bigger ones on there, if not the biggest out there now for aquaponic cannabis. So definitely check it out. DP Davis asks, the city water is 450 to 500 ppms. Can I use rainwater instead for my hydro system, or for his aquaponic system? My rain barrel is below 50 parts per million. So this is an interesting question. So total ppms means almost nothing in aquaponics. If it's super high, it can mean you have a lot of calcium in the water or something like that. But aside from that, it really means absolutely little to nothing in aquaponics, because it doesn't tell you what those nutrients are. What I would recommend to do is to take that city water and have it sent out for an agricultural or mining water analysis. Depending on where you are, it can be anywhere from $40 to $100. And then find out exactly what it is that you're high in parts per million of. And that's any grower, any gardener should be doing this, so that you know how to adjust your own nutrients based around what your tap water is heavy in. If you already have a shit ton of calcium, you're going to cut back your calcium dosing, because if you're using tap water, you don't need it, or you might need to put a stripper on there, to lower the pH before it comes into the system or something like that, depending on what you're doing. So, getting a baseline and understanding what those ppms are, is it 200 parts per million calcium and 40 parts per million magnesium and whatever else for the rest, or is it all cyanide, or is it aluminum, or is it whatever? Who knows? And this is how I've gotten to some of my more ridiculous questions. People ask me, like, what's the allowable uranium level or radon level in your water, and all kinds of stupid stuff that you should never ever try to grow in based on their water results. So it's also good to know for your own personal health, what you're drinking, so that you can take appropriate action as well. I mean, look at Flint. If someone had done that a while ago in Flint, they would have picked up on that shit.
If you're in a city area where it's city water, it's been proven that a lot of cities, you're getting drugs that have been flushed down the toilet or whatever. I mean, any kind of freaking like pharmaceuticals could be even part of your water, parts per million. So best thing to do is use RO water. Right, Steve?
I don't know. I use good
that or
well
rainwater.
water. Well water is good if you have good well water. The other one I like is cold sterile. If people have done planted aquariums, there's a method called cold sterile, KOLD, where you can strip out a lot of the heavy metals and a lot of the like a portion of the alkalinity without totally stripping it completely out. And that's basically like a part of the RO filter with part of it taken out of it. I forget. I think it's just the carbon removed. But it's not exactly the same, but it's similar in concept. It's a resin matrix that you run the stuff through. So that's what I guess I would recommend if I had to.
Can I get back to the question one more time? The original question?
The original question is, my city water is 450 to 500 parts per million. Is this too high for me to use in aquaponics? And that very well might not be too high. It just depends on what it is that makes up that base.
In regard to his ask, he said he has rainwater that was 50 parts per million. I would say yes, definitely you can use that.
Just remember you have pH and alkalinity problems with super low.
That's what I was going to suggest. You have to check and see what your pH of your rainwater is. That's the most important part of the answer to your question is what is the pH of your rainwater?
Well, rainwater will always be 7 unless it's super pollutant near you. And then it'll slowly, the pH will crash over time as it sits there.
I see people all over, they say their rainwater, they collect rainwater and their pH is all up and down the scale. You think, you have to have a perfect situation to create neutral rainwater, I think, or you might be right. I mean, so acid rain, I don't know. I mean, there's a rain is different, right, Carlos? All rain is different.
Well, I've actually been doing a little bit more research on how people harvest rainwater, and there's actually, like the guy said, his ppm was as low as 50 for his rainwater. Mm-hmm. And I know people do what's called a first flush diverter, which is one thing I'm working on doing. I'm trying to get a full filtration system for my rainwater so I can have really clean rainwater that goes into my system. I would just recommend making sure you have something that you have to keep your pH in check. I mean, because especially with aquaponics, it's something that you can't just be letting go all over the place anyway. Otherwise, your fish are going to suffer for that too. So, I mean, pH is generally something that a responsible aquaponic grower keeps up with anyway.
It all starts with the pH.
So it turns out I was wrong. pH of rainwater is actually 5.7. And it has to do with, it starts off at 7 when it's formed in the cloud, but as it falls to earth and reacts with the carbon dioxide in the atmosphere, it lowers it temporarily forms carbonic acid within the rain and lowers the pH.
So it could be, but
yeah, I was going to say, I'm not exactly sure what that has to do with, but I know whenever I get my rainwater, it's usually at about 6.5.
So acid rain is when pollutants in the nearby atmosphere generate a pH that is, 4.9 or below. Anything in the 4s.
That's a pollutant like smog and stuff. If the rain's coming through the smog, it picks it up. So here's the interesting thing. Since this question, I like to say this. I have just started collecting rainwater for the first time ever. I let it rain all over me and all that. I just started with all my experiments. I found my 5-gallon soap barrel, and I've rinsed it out real good. And I'm going to fill it up and dump it out, fill it up and dump it out, and I'm going to start collecting rainwater. And I'll let you know what I get, as far as...
yeah, I will say it's definitely not as simple as you would think. And I feel like we can probably extend this conversation a little bit more on ilovegrowingmarijuana.com, because I'll throw some links up and whatnot so people can check that out. Because if you're going to be doing rainwater, like the first thing that you have to think about is you have to keep mosquito reproduction down. So you have to have screens, obviously, that's like the most obvious thing. And then on top of that, the one thing that I only came across after I thought about it more, and I was like, there has to be some kind of solution for this, and which there is. Like I was talking about that first flush diverter, like you basically just have some sort of reservoir or pipe or something like that that holds a certain volume of water. I think you can actually get an exact measurement for how much volume you need to hold based on the square footage of your roof. But what that does is it'll basically take on all of the first material that gets washed off your roof from the rain, and then once that fills up, everything that falls into your rain barrel is, you know, fairly clean to start with, as far as the debris and whatnot. And then from there, like I was talking about a little bit on the episode last week, a lot of one thing that I've seen that I'm going to do, of course this is going to be a little bit later down the road before I make a larger scale one, but I've already got my little filter set up. I just got to get some better sand, because the sand that I had for it was apparently way too fine and wasn't letting hardly any water through. But you have basically the same sort of biofiltration that occurs in aquaponics, only it's for pathogens and whatnot. You will form a biolayer on the top of the sand that will actually eat pathogens and other types of harmful or unwanted microbials. So, and then that will, like whenever you take your rainwater without any kind of filtration, even a first flush diverter, you can, that tests up to like 300 or higher colony forming units of bacteria or CFUs. And on one of the channels that I seen, this guy had his water tested that comes out of a sand filter that's been functional for like several years, and that comes out like super low, like less than 10 colony forming units of bacteria. So, and then if you're worried about microbes at all anyway, you can just, at that point, for drinking anyway, you can just boil that off, and you know the sand has already taken any kind of particulates out of the water, and it really comes out super clean. So that's one thing I'm looking into scaling up on here in the near future.
Well, that's awesome, because there's so many people on the site that would love to have somebody like you talking about really is experimenting with and controlling rainwater. And
Yeah.
we all have ideas about it, right? And I've got, well, see, I collect my rainwater straight into my receptacle. I don't use a roof or anything, so it's a little different too. So that's different too. But one, thank you for mentioning the site, and thank you for both of you guys joining too. I want to really thank you both. We got to get that podcast posted up there and let the members start, checking it out. Some of them go, it's too long. I'm sorry, Steve. I think you wanted to talk again, and I jumped on you again, didn't I?
No, it's fine. It's some people say the show's a little long, but this is what I liked about it is, or the reason why I like the current format. If you guys feel super strongly it's too long, we could chop it up or something. But
I don't think we're talking about really good information right now. This is when it should be long.
Yeah.
Yeah. I mean, I feel like as long as the information is informative, people will stay watching,
Yeah.
Yep. We've had 6 people already that have been watching the show since we launched, so it's pretty awesome.
Nice.
Yeah. I mean, that's this whole, 2 hours now it's been? So.
Oh, we still have 6 in chat?
Yep. So.
Thank you, guys.
Hell yeah.
I tell you what, this is one of the best shows we've had in a while.
Oh, hell yeah, it is.
Mammoth P was badass, man. I mean, what a PhD. How often do you get to see a PhD that'll sit there and be like a homeboy, a good old boy, the guy out there, you could meet him on the beach. He probably surfs.
Yeah, that was fun. That was cool. Me and Marty were whittling away at his secrets, or trying to anyway. That was every time Marty would ask a question. Like Marty threw out the first question that I wanted to ask. I was like, all right, Marty got there first. Cool. So then I was sitting there writing down the first question I asked him, kind of went along with that, and then Marty kind of went and asked another question related to the question I asked. So I was like, all right, cool. We're trying to get to the bottom of this. So it was pretty cool. It's always fun having a really informative guest like that.
Oh yeah, he was awesome. I tell you what, and I got a lot of people I need to know for sure about the show, if it's next week or whenever the big LED shoot off is. I need to know that because I got tons of people that well, Steve, you can post it, but you need to build it up, man. Build it up on the forum.
Yeah, I mean, if that's gonna be next week, then
Make
for
them
sure.
make them want. Well, if you put something out there yourself, I'll go out and I'll say about it, and we'll get some other friends like Chicken Hole there, who's not Chicken Hole on the forum, but he's my buddy. So, and he loves the show. He loves learning shit too. We all love learning. And anyway, we'll rock that thing. We'll roll it up into something because we got a great. I don't know how much time you really spent there, but we've got a great membership. And if we promote it right, there'll be a bunch more people coming in here because there's a bunch of people. I keep telling them, and it's not all aquaponics. Just like we talked about rainwater, and Brain Grow had all this great information about rainwater that, like, if I was like really wanting to use rainwater, and if I said, wow, which I'll review this show as, there's an excellent discussion at like 2 hours and 20 minutes or whatever it is that is about rainwater that you might want to listen to. I think the one thing we might do in the show, you brought up shortening it or whatever, when we post a podcast in a network type situation instead of the actual live, we might want to make them abridged. And then like sometimes, because, well, not all shows, like tonight I wouldn't abridge anything. But there's other shows where we just go nuts or go, nuts, and it's like, okay, well, where does the stoner moment for like 10 minutes, So we could get rid of that when we put actual recording out there for like, listen to show this or show that. We could get rid of some of the stuff that's wasteful.
I just try to put on something that like people can listen, put on while they're working on their plants in their grow room. I like to spend, when I have a personal grow, I like to spend an hour working on my plants, maybe 3 times a week, 4 times a week, cutting clones, pruning, LSTing.
I like that.
Whatever. And
that
it's
way
nice just to have something to put on. That's why I do the Do Grow show. I'll put it on while I'm working on the plants or working in the garden, or especially if I'm working, on a project or something. It's cool because like other people are learning stuff that they don't know too. We're all kind of listening and learning, and it's kind of how what I consider the format for this show is kind of something to put on while you're working on your grow.
Oh yeah, and I wasn't disputing that. I mean, I love it. I'm here every week. I love it.
Awesome. Yep, and so
it is awesome.
Yep. So guys, make sure you check out our shows. We have Growing with Fishes on Thursdays, and now we have Last Week in Cannabis on Mondays. It'll be Monday or Tuesday. Some days it'll be Tuesdays, depending on the week. We'll try to give you guys as much heads up as we can. I know we just switched it up this week. We were going to try and do it Tuesday, ended up doing it Monday anyway, but we're going to try and keep it on Mondays, at least for now. We'll see how that works out longer term schedule wise, especially once I get settled here up in the Bay Area. So I think that's it. If you guys are interested in learning more about us, check us out. There's a link to everyone's YouTube channels in the description as well. Be sure to check that out. What else is there? I think that's it. Check out my YouTube channel, Potent Ponics, on YouTube. You can check us out again. We have the Facebook group, Aquaponic Cannabis Growers. I also have potentponics.com. I uploaded all of our stuff to SoundCloud now. So if you're interested in listening to us in an audio format, I'm working on back cataloging all the old episodes up onto SoundCloud now. I also have the RSS feed that's set up on iTunes. I'm having a couple of problems getting the RSS to update everywhere. Hopefully I can figure out what the hell's going on with that. But I'm working on getting us up and hosted in much more places now, with the SoundCloud and getting the RSS up and everything, and trying to expand the show. So if you guys know anywhere that's cool to share the show, this would definitely be a great episode to go out there and share that. So if there's, you guys know communities of people that you're involved with, and you think they might enjoy this show, please share it with them. Thanks, everyone. Also, shout out real quick to Ouroboros Farms. I'll be having a little bit more content from there because I'll be living not too far from there, so I'll be able to do a lot more content with them in the future. We'll have a lot more stuff with them too. So shout out to them. Check them out. They have a bunch of different classes. I was just up there for a farm to table with them on Saturday. This past Saturday, it was really cool. They had a wonderful farm to table that they did with a local community and a local chef. And they had, I think, everything from that farm was within 10 miles. Or everything from the dinner was within 10, grown or slaughtered within 10 miles or something like that of the farm. So that was really cool. And yeah, I think that's everything for this week. So thanks a lot, everyone, and big announcements coming soon. And we'll see you guys next week. Cheers, or I guess we'll see you on Monday. Take care.
Good night.